Good morning, everyone. Welcome to our 2022 presentation of research results from our Research Experiences for Undergraduates program. For those of you who don't know me, I'm Rochelle Seitz. I'm the director of the program. And today is one of my favorite days of the whole summer because we get to see everything that the students have been working hard on for the past 10 weeks. And it's always very gratifying. So I'm really excited to see that. But before we get started with that, I wanted to just take a little trip down memory lane and talk a little bit about some of our group experiences. So let's see if I can get this going. The interns arrived in late May and we were starting to bond a little bit. We took our trip to the Eastern Shore and our first stop was at the Cherry Stone Aqua Farms, where we saw their clam and oyster operations. And a few of us got to taste some oysters, sometimes some of us for the first time. So that was pretty exciting. How'd you like it, Anissa? Your favorite? Okay, we're not going to tell. And then we made our way to the Eastern Shore Lab. We had some great boat outings and some nice meals cooked in the dorm. We went out on the boat. We had a chance to see some of the great birds they have out there, a nice bird rookery. And of course, we had to collect some plankton and check the water clarity, getting some use of new equipment for some people. And one of my favorite things is always playing in the mud. Everyone got nice and muddy, dug a huge hole and got some good bivalves. So you guys did a great job out there. I always say the more muddy you get, the more fun you're having. So. And of course, we found some really cool stuff, getting close to the animals. I don't know if you actually kissed that horseshoe crab, Cole. That might have been a little prickly, so good decision. And then, of course, the beach seining was always a highlight. Everyone got in there, a lot of good teamwork, got some good samples, got to see some cool fish and crabs. And we also did some scraping in the creeks. Some other things that lived on the hard substrate, like the sea urchin was a big, one of our favorite species, I would say. Then we also did some trawling in the creeks, got some more crabs and a couple of new species of fish. And here's a few of them that we got, a couple of pictures. Everyone wanted to take a picture with the crabs and cute little oyster toadfish, cute and ugly at the same time. Brought everything back to the lab, did some looking and IDing in the lab under the microscope, and saw a couple of things over in that tank. It was nice to watch them swimming around, and the whelks were climbing up the side. There's our group in front of the seawater lab. And another field trip we had to the tidal fresh waters. This was, I thought, was a fantastic day to be out there. Some people canoeing for the first time, so learned a new skill. That was nice. I guess it was a hot day, but I think we had a really good time out there. And Jim Perry was out there teaching us all about the flora and fauna. That was great. Had another field trip on the Bay Eagle, and this was the first time in 3 years that we've been able to go out on the Bay Eagle. So it was great to get out there, and I think everyone enjoyed a nice day out on the water. And we did some collections. Again, we did some trawling, this time in the York River, and we had some help from Mike Sebo and Alex Snyder, and also Paul Richardson is in the upper left there. And of course, the boat captain, Bubba Hogg, was there. And we caught some cool fish, got one there, Cole, in the lower right with the parasitic isopod in the mouth. And of course, we did some plankton sampling. We also had some help from Maya Thomas, and everyone got into the jellyfish. Good thing you were wearing gloves. Lots of jellies out there, particularly in the upriver station. And my favorite is always sampling the benthos. So everyone got into the mud. We got some really good silky mud and really good for face painting. So that worked out well. And here's our crew at the end in front of the Bay Eagle, just after a fun day out on the water and hopefully educational day as well. So then after all those field trips, everyone got involved in their own research and spent the majority of their time on their own projects. So that's what we'll hear about today. And so the field trips were really just the beginning of our REU 2022. But before we get to the student presentations, I wanted to give a big thanks to Stephanie Peart and Grace Massey. So a round of applause for our TA and program coordinator. Without you guys, none of this would happen. Seriously. So thanks so much. And I also wanted to take this opportunity to say a thank you to all the mentors. So a round of applause for all the mentors. Again, you know, we really appreciate all the help that you've given the students over the summer. And again, couldn't do it without you. So with that, those were a little bit about our group field trips, and now we want to hear about the hard science. So the way we're going to do it is we're going to have 2 sessions, and each will be moderated by one of our interns. So our morning session is going to be moderated by Sarah Vedatcher, and she's going to introduce each of our speakers, and they will each have a 20-minute time slot, 15 minutes for the presentation. And we'll give our moderators will give a signal when you have, are we going to do 1 minute left? Or when you have 1 minute left, give you the 1 minute sign. And then at 15, we'll give you the cutoff sign. And then we'll, excuse me, have five minutes for questions. And again, I hope all of you interns will be asking each other questions so we can have some good exchange at the end. So with that, I will turn it over to Sarah. Oh, one more thing for those of you on Zoom, we're going to do Q&A. If you can please type your question into the chat, and we'll go from there. And Stephanie will read the question, and then the speaker will be able to answer. So now I'll turn it over to Sarah.
Hello, everyone. So we'll be starting with Eli Whitehead-Zimmers, and he will be talking to you all about sediment transport, and he is from Messiah University.
Good morning, ladies and gentlemen. My name is Eli Whitehead-Zimmers. I am a senior physics major from Messiah University. And this summer, I've had the immense privilege of working with Dr. Courtney Harris on our project, Duel of the Fates of Ayeyarwady sediment. Does any sediment make it into the Bay of Bengal, or does it all stay trapped in the Gulf of Maribor? Well, you may be asking, what's an Ayeyarwady? Great question. It's a river. Here you can see in Southern Asia. Zoom in on this dotted box. And welcome to the country of Myanmar, where we have these 3 rivers, the Ayeyarwady, the Setang, and the Salween rivers, which make up our Ayeyarwady River system. Now, this system each year puts 485 megatons worth of sediment into the ocean. Now, that's important because ocean sediments are the #1 reservoir of organic carbon in the world. In fact, this river puts 6 megatons worth of organic carbon every year into the ocean. That's the 2nd largest in the world. Now, it's important where the sediment ends up. There's 2 main depot centers for this system. One is in the Gulf of Maribor here to the right, and on the left, the Bay of Bengal. Now, the Gulf of Maribor is very tidally active. It has a tidal range of over 4 meters. Additionally, it's sensitive to what's called the spring and neap tidal cycles. Here you can see a diagram that when the sun is in line with the moon, we get these larger tides called spring tides. Whenever the sun is at a 90-degree angle with the moon, we get these smaller tides called neap tides. So this Gulf of Martaban, very tidally energetic. It's mixing up a lot of sediment, resuspending the sediment into the water column. So the organic carbon in the sediment has this greater chance for what's called reoxidization, or the carbon is actually being put back into the carbon cycle. In the Bay of Bengal, however, it's not as tidally energetic, and there's more of a chance for the carbon to be sequestered or taken out of the carbon cycle. We call this kind of area a carbon sink. So how does sediment get to each of these two depot centers? Well, in this project, we're going to focus on three main delivery mechanisms: the winds, the waves, and the tides. Now, seasonally, these three delivery mechanisms are influenced by monsoons. In the summer, we have a southwestern monsoon. It's very rainy, it's energetic, and it's windy. We can see this pink arrow shows the direction of winds during the southwestern monsoon. In the winter, we have winds coming from the northeastern monsoon, shown by this blue arrow. It's a little bit drier, it's not as energetic, it's less rainy. But in the literature, it's been thought for a while that these winds from the northeastern monsoon cause these blue surface current arrows. And these surface currents would carry the sediment from the Ayeyarwady River system into the Bay of Bengal. Recent studies, however, have shown that the sediment in the Bay of Bengal is more indicative of rivers stemming from the Indo-Burman mountain range. So the question is, does any sediment actually make it into the Bay of Bengal? This study set out to answer this question of estimating the flux to the Bay of Bengal. Additionally, we want to determine significant mechanisms of delivery into the bay, namely the wind, the waves, and the tides. To do that, we're going to use a three-dimensional transport model developed by Matt Fair, who's a VIMS graduate. Here you can see a picture of the model output, and we can see these different blue dots represent different river inputs into the system. This is a red transect. This line is the transect from which we're going to measure sediment across. The idea being, if we see a lot of westward flux, it alerts us that it's sediment's moving into the Bay of Bengal. We're going to run this model for a northeastern monsoon because that's what the literature tells us we should expect this flux westward into the Bay of Bengal. We're going to run it for the month of January, but specifically, we're doing analysis on January 14th to the 28th, a spring neap cycle. Now, we're doing this to allow for 14 days of spin up to let our model adjust and get acclimated. We're going to do two types of runs with this model. We're going to have a baseline run and several isolating runs. This baseline run is going to have realistic winds, waves, and tides, and we'll use it to estimate the amount of flux to see if we get any sediment into the Bay of Bengal from the Ayeyarwady. We also have a series of isolating runs. So let's say, for example, we're isolating the winds. We would turn the winds off, but leave the waves and the tides as realistic. Then we'll estimate the flux through our transect and compare the flux to our baseline case. And if there's a huge difference, let's say when we turn off the winds, we can say, okay, we turned off the winds, we saw a big drop in sediment flux. The winds must be responsible for transporting a lot of that sediment. So in all, we have four different model runs: the baseline case, the no waves case, the no winds, and the no tides. Here you can see diagrams showing the different forcing that the model uses, basically how the model represents the winds, the tides, and the waves. On the y-axis, we have the wind stress, the wave orbital velocity, and the tide magnitude. And then I have plotted the baseline, no winds, no waves, and no tides case. What's important about this slide I want to show you, however, is the tidal magnitude. You can see here, we have the spring tide, a large tidal range, going to the neap tide, a smaller tidal range, back to the spring tide again. Before we dive into the flux results from this study, I want to show you this really cool animation that gives a good intuition for what this model is capable of. Here you can see these arrows represent the bottom surface current, or the bottom currents, and the brown coloring shows the sediment concentration on a log scale. And again, let's focus your attention to our red transect here on the left. Let's take a look. Wow, already you can see these currents are moving back and forth along this transect, affected by the tides, going east, and west. Additionally, you can see some brown splotches moving down from the north moving southward along this shelf. Interesting. So we can see that this system already we're getting a feel. Tides are a big player here. Okay, let's investigate a little bit further. Let's zoom in on our transect. Specifically, let's take a look at the cross section of what's going on here. Let's investigate how much flux is going through this transect and how the sediment's moving through it. The red colors in this animation are going to denote eastward flux, and the blue is going to denote westward flux. Let's take a look. Wow. Again, we can see tides are moving this sediment back and forth, east, and then west again. The tides are a big player that are involved in moving the sediment across this transect. Specifically, I want to show you that whenever it's going westward, we have the sediments actually in this deeper water. And whenever it's going eastward, the sediment flux is happening in the shallower water. Interesting. Hold on to that. Put that in your back pocket. We're going to come back to it in a second. But if we were to distill that animation down into a time average, we can see that on average, we actually have a sediment flux going eastward. Well, why is that? Well, if we take a look at our velocities going through this transect, we see the westward velocity in the deeper and the eastward velocity is in the shallower waters. But when we look at our sediment concentration, we can see that on average, it's actually in the shallower waters. That's where the eastward current's moving. So that explains why we see this eastward flux in the shallower water. While we have an average flux towards the east, I want to stress again, this is a tidally influenced system. And there's really two different ways you could sort of think about this. You could think of it, one, as sediment is just getting whisked back and forth across our transect. And slowly but surely, all of our sediment is sort of inching eastward so that all of our sediment has moved east. That's one way you can think about it. Second way is that we have sort of a sediment highway, if you will, where there's a continuous exchange of sediment between these two systems. Near the coast, we have sediment moving eastward, and it keeps on going eastward clear to the Gulf of Martaban. Or when we have sediment in the deeper waters, we have it going west, and it keeps on going west to the Bay of Bengal. Now, the data would seem to suggest the second sediment highway, because when we look at our currents, again, we see these two very distinct channels for transporting sediment. And remember, back from those clips I showed you from the animation, take it out of your back pocket. When we saw the flux going westward, it was in the deeper waters. And when it was going eastward, it was in the shallower waters. Seems to suggest this continuous exchange of sediment. Now, if we were to take what we saw in that video and plot it as a time series, we could look at the flux for each day of this model run. So here we have the sediment flux in kilotons per hour for each day during our model. And the positive is eastward flux, the negative is westward flux. And again, we can see this tidal variation, east, west. Now, we had that average flux going eastward. That number, we actually have a net flux of 537 kilotons going towards the east. If we take a look at this dark purple line, this is a moving average of our data. We can see this interesting sort of bulge of eastward flux during the middle of our 2 weeks. That's weird. Let's take a look at why that is. Well, when we look at it with our spring neap cycle variation, we see the spring tide here with this larger tidal elevation is when there's about an equal east and westward flux. But as we move into our neap tide, we see this almost exclusive flux eastward. And then to the spring tide again, we see it goes back to being equal. So not only do we have tidal variation every day, but on this larger 14-day time scale, we have variation with the spring tide cycles, the spring and neap tide cycles. Very interesting. Now, if we're going to operate under this sort of sediment highway model, this metric of net flux isn't very useful to us. We want to know how much sediment is moving east and how much sediment is moving west. So let's take a look at that. Here I've plotted the cumulative sediment flux. Cumulative just means that each day is a sum of all the other fluxes before it. And we can see here, this is our baseline. We get this spike of eastward flux from the neap tide, and it levels off to about 537 kilotons net flux. But if we look at just the eastward flux, we see we actually have 700 kilotons worth of sediment going eastward. Likewise, we can look at just the westward flux, and we see that we have 150 kilotons worth of sediment moving westward. Now, if you were to look just at the net flux, you would say, oh, well, we only have 537 kilotons moving east. That's not the case. We actually have a significant amount of sediment moving both directions. Now, all throughout this baseline run, we've heard this story again and again. Tides influence the flux. When we take a look at our isolating runs, we see the same story. Here I've plotted the cumulative flux as a bar graph for each one of our model runs, baseline, no winds, no waves, and no tides. While the scope of this project didn't allow for a statistically rigorous evaluation of the variance between these 4 different cases, we can see graphically that the no winds and the no waves case aren't that substantially different from the baseline case. The no tides case, however, you can see the net was cut by over 300 kilotons. Additionally, when we look at the westward flux, we can see that it was cut more than in half from the baseline case. So we see the tides are important for transporting sediment, but especially so for sediment moving westward. This project used a 3-dimensional transport model to approximate the amount of flux going into the Bay of Bengal. We discovered that there's actually a net transport going to the east, but we also discovered that the question of where does sediment go is a little bit more nuanced than looking at just the net flux. We propose a sort of sediment highway model between these 2 systems, where there is a continuous exchange of sediment between the Bay of Bengal and the Ayeyarwady River system. Additionally, we discovered that one of the most significant delivery mechanisms within the system are the tides, especially for moving sediment towards the west. These novel results introduce a lot of new ideas into the literature that are going to require further investigation. It's our hope that these results can be used to inform future carbon budgeting studies for this system, so that we can better understand the Ayeyarwady's role within the global carbon cycle. I'd like to take a few moments to thank all the people that got me here today, especially Dr. Courtney Harris. It's been an amazing summer. I've learned so much. Evan Flynn and Dr. Stephen Keel helping me interpret results. Dr. Dongxiao Ying for all of his help throughout the summer, coding, looking at my papers, interpreting results. NSF for funding, our REU program advisors, and my REU cohort. Thank you so much, everyone. I'd love to take any questions.
Okay, so now if anyone has any questions, Stephanie will run around and get to you. James.
Great talk, Eli.
Thanks, James.
Could you go back to the graphs of your isolated runs?
Yeah, this graph.
Yeah.
Mhm.
So I was wondering, the winds and the tides are pretty heavily interconnected. So when you had like no winds
Mhm.
and you did that run, did that factor in how the tides would change also, or was that not something you were able to factor
Yeah.
into the model?
Good question. So if we go back to, you can see this slide of the different forcing. So this is what the model used for the winds and the waves and the tides. So you can see that, if we were to look at the no winds case for the wind stress, we see, okay, the winds are 0. But if we were to look at the winds for the no tides case, we see that it closely kind of mirrors the other runs. Good question. Thanks, James.
Hi, I have a question about validating your results.
Yeah.
Do you know of any precedent or whether it's standard to have sort of a field experiment, maybe to assess like where the sediment transport is coming from, maybe through isotope ratios or something like that?
Yeah, great question. So some of those recent studies I talked about in like where the sediment's coming from in the Bay of Bengal, Evan Flynn is one. She just did a paper, published it this year. And she went and studied like different sediment types. And they were the ones that kind of figured out that, okay, maybe the sediment in the Bay of Bengal is more like it matches more the type of sediment you would expect to come from the Indo-Burman mountain range, just smaller rivers. So yeah, there have been studies that go on over there. It is rather an understudied system, however, due to the political climate, a lot of like funded studies that were meant to go over there and collect more data to kind of validate studies like these had to be postponed or canceled. So we're kind of working with what we got from things that have been published the last few years.
Really quick, we got a question from Zoom audience. They say, great talk, Eli. Can you speculate on the role of large storms on the net east or west transport over a longer time scale?
Yeah, that's a great question. And in the literature, doing the background research for this, that's definitely it could be a big role for transporting sediment. The idea is you get like a big storm or cyclone, and that could just mix up your system. Like this model kind of ran under normal conditions. But if you were to get an extreme energy event, the idea is that maybe we could get a sort of big pulse of sediment from the Ayeyarwady system into the Bay of Bengal. And a lot of the literature seems to point that that could be a big contributor to the sediment types that they find there.
Hello. I have kind of a random question, but I
Sure.
was wondering the importance of sediment deposits. Is it just like geology, like where it's located, or is it gonna have like aspects on like the organisms within the water, any minerals or?
Yeah, great question. So question is like, what's so important about where the sediment is deposited? Kind of touched on that in the introduction. The idea is that if it's in the Gulf of Martaban, the organic carbon that's within the sediment, if it gets mixed up a lot, you're releasing a lot of that carbon back into like the atmosphere. Whereas over here, it's going to get sequestered. Whereas like the carbon is just going to be stored at the bottom of the ocean. So the idea is you could do like what's called like a sort of a carbon budget for this system and say, okay, what's sort of like, are we getting carbon output from the system? Is it a carbon source or is it a carbon sink? Is that actually storing it? And especially for climate change, we're really, wondering where carbon goes, why is it getting stored in the atmosphere, things like that. If you do sort of a baseline study, you can measure metrics of human impact. So if they start damming the Ayeyarwady system, they start deforesting that area, is that going to impact, is it going to become a carbon source now instead of a carbon sink? And how does that play into the grand climate cycle and especially impacts for like climate change? Good question. Thank you.
Then I think we have time for one more question, if anyone else. All right, then thank you so much, Eli.
Thanks, guys.
Never- So our next presenter will be Sophie Galvin, and she is from Frostburg State University.
Thanks for the introduction, Sarah. Great talk, Eli. It's going to be a hard one to follow, but hi, everybody. Again, my name's Sophie, and this summer I had the pleasure of having Dr. Bill Walton and Julia Gren as my mentors, working on a project that aimed to understand the effects of stocking density and biofouling control practices on the biofouling growing on oyster bags. So aquaculture is considered one of the fastest growing industries for food production worldwide, with marine bivalves accounting for 60% of coastal and marine aquaculture production. Especially here in Virginia, where shellfish aquaculture continues to grow significantly and provides notable economic value to Virginia's marketplace. Since it is the #1 producer of Eastern oyster here on the East Coast, and it supports a more than $30 million industry in conjunction with the state of Maryland. So not only does aquaculture have economic value, but it's also ecologically beneficial, especially when it comes to shellfish, because, for example, oysters provide various ecosystem services, such as improving water quality, and even helping with the sequestration of carbon. Interestingly, around the year 2012, oyster farmers began reporting high summer mortality rates nearing 30%, and even in some cases reaching 85% mortality. These incidents are not directly related to disease or other environmental stressors, such as algal blooms. Which leads me to believe that biofouling could be a potential stressor, especially since it tends to reach its peak during the summer due to increases in productivity. So what is biofouling? As you can see from my picture, which is of an oyster cage that has a mesh bag in it where you're supposed to be seeing oysters through it. It's just the accumulation of various marine organisms on submerged structures. So since we can see that from this picture, it's completely covered, you can't see anything. It can cause various detrimental effects, especially on oyster growth and survival, where the biofouling grown directly on the oysters can impede their movement and affect their growth and shell condition. Also, the biofouling communities can colonize the outside of the bags, as we can see here, which can create blockages that impede essential water flow. So all of these things combined can contribute to economic losses, as it's estimated that 5 to 10% of aquaculture production cost is directly attributed to just biofouling control. So the objectives for my project were to, one, identify the fouling communities and quantify them, as well as determine whether stocking density and biofouling control practices had any effects on biofouling. My null hypotheses were that I expected to see no differences in biofouling measures between inside and outside plates, no differences between air-dried and non-air-dried treatments on both plates and the grow bags, no differences in stocking density treatments, and no interactions among stocking density and air-dried treatments. So the study area for my project this summer took place at the Big Island Aquaculture Farm here in Gloucester, which is represented by the red star on the map. I should also clarify that my experiment this summer stems from a larger graduate study that's looking at how various metrics, including stocking density and air-dried and non-air-dried treatments, are affecting the microclimates within the oyster bags. And as you can see from this picture, this is exactly how the bags were set up in the field, where there were 36 bags with empty, normal, high stocking densities with corresponding air-dried and non-air-dried treatments. So part of my methods this summer were to collect biofouling and stocking density data on 3 different farms that we hope will add to what we know about biofouling across a variety of oyster farms in Virginia. However, for the sake of time, I will only be focusing on the data I collected at Big Island Aquaculture, which involved the collection of biofouling using these small 3x3 settlement plates that I created that I cut out from oyster bags, that I then attached to both the outside and the inside of each experimental bag. These plates were left out in the field for about a month, and after I collected them, I assessed the biofouling communities, as well as photographed all of the plates to be able to process them through ImageJ and determine the % open area, which is just another metric for measuring biofouling. I also took the wet weights of the biofouling, which again is just another way to measure the biofouling that accumulated on the plates. And after all of my data was collected, I conducted various statistical analyses, such as a 2-tailed pair t-test, just to see general differences between the inside and outside plates, a 2-factor ANOVA to determine whether there were any differences among treatments, and a 2-key test to see where those differences were happening among the treatments. So for my biofouling communities results, there were not a lot of taxa present, which I assume was because the plates were not left out in the field for a long period of time. So, but what we had in some of the most prominent organisms that I saw on most of my plates included tubeworm fouling, different types of algae, such as brown algae, green algae, two types of bryozoans, such as the cushion moss and the invasive encrusting colonizing bryozoan. As you can see from this picture here, or the bottom picture, they can completely cover everything they settle on, which is a problem for oyster bags. Some of the other taxa I found included barnacles, the Atlantic paper mussel, sea squirts, and my favorite, anemones. I know this isn't a very exciting picture because they're out of the water, but I thought it was a really cool find. So yeah. Now moving on to wet weights, looking at just the inside mesh plates, where the x-axis represents empty, normal, and high stocking densities. The y-axis represents biofouling weight in grams, and each letter represents significant statistical differences between groups. So as we can see from the empty non-air dry treatment represented by the letter A, it had the greatest significant differences when compared to the rest of the treatments, regardless of biofouling control and stocking density, which means that an empty bag that's not being frequently air dried is showing the highest amount of biofouling accumulation. Moving on to the outside mesh plates, there were no significant differences among treatment groups except between air dried and non-air dried treatments. And we can see that there were statistical differences where the air dried treatments again showed the least amounts of biofouling. Now we're talking about percent open area, which is a little bit of an inverse from my previous graphs, where now the highest number essentially, or the highest percentage represents the least amount of biofouling on each plate. Again, we can see that the empty non-air dry treatment, when compared to the normal densities, had significant differences. However, when compared to the high stocking densities, they appear to be similar, especially since the high stocking density values were intermediate. Looking at the outside mesh plates, 4% open area, we can again see that the air dried plates, for example, when looking at the top clean plate up there, had the highest percentages of open area, which allowed more flow into the mesh bags. So that's just another way to look at it when compared to the more biofoul plates. So now moving on to total bag weights in kilograms. I should note that these bags were deployed or have been out in the field since May, show similar trends as when compared to my plates, that just looking at the air dried treatments, regardless of stocking density, we can see that they had the least amount of biofouling. However, when looking at the non-air dried treatments, despite the amount of biofouling on them, we can see that as stocking density increases, the biofouling decreases, which is interesting. So just to summarize everything I just mentioned, I saw no significant statistical differences between inside and outside mesh plates. Air drying was in fact an effective practice, as you guys saw, and stocking density did play a role in the reduction of biofouling, especially when looking inside of the bags, which is interesting because the more oysters there are in a bag, you'd expect that they could act as substrates for fouling communities to attach to them. So seeing the opposite is kind of cool. So some of the reasons we think this could be happening could be due to competition for food, incidental predation, where the oysters are unintentionally filtering the larvae of fouling communities or fouling organisms, and or the physical interaction with the oysters where they could be rubbing off the biofouling from each other or within the bag. So my project This summer will contribute to the ongoing graduate study that will hopefully could potentially address whether biofouling is in fact having water quality or is affecting the water quality within the oyster bags. It could also inform oyster farmers by providing them with better data so that they can make more informed decisions, which will in turn hopefully improve the relationship between academia and industry through mutual collaboration. I would like to thank my lab, my mentors, Dr. Bill Walton and Julia Grant, as well as David for teaching me everything about ImageJ and always being there to answer all my questions. Jordan for helping me out in the field, and Jennifer Dreyer and Shannon Hood for helping me identify some of the fouling taxa. I would also like to thank the VIMS REU coordinators, Dr. Rochelle Seitz, Dr. Grace Massey, and Stephanie Peart for allowing me to be here. And my funding source, the National Science Foundation. Any questions?
All right, so we have time for a few questions. I think we saw, all right, Eli.
Thanks. That was an awesome talk, Sophie.
Thanks,
I was
Eli.
wondering if you could describe a little bit more in detail what all air drying entails. I'm just thinking like if they were just like air drying outside, would
So-
like the sun be like a factor in sort of like killing some of the bacteria, or what was that like?
Yeah, that's great. So air drying is literally what it sounds like, as you can see from that picture. Literally all you do is put the some bags on top of other bags so that they're exposed to the sun and then not actually touching any water so that the organisms on them can die, really, yeah. Mhm.
Hey, Sophie, excellent talk.
Thank you.
How often were the bags dried and for how long?
They were dried every 2 weeks. So I guess that answers your question, which is more than people than oyster farmers do. I believe I was told that some farmers actually don't do air drying. They might just do air drying for maybe like once every 2 months, which is not a lot, which is why in some farms you can see that their cages or their bags are just heavily covered in biofouling. I hope that answered your question.
Hi, Sophie. You did the absolute amazing best job, and I love you. Anyway,
Thank you.
What was your absolute like favorite thing or like organism that you found with bags? Like your favorite biofouling?
So again, like I said, the anemones were really unexpected. I didn't even know what they were when I saw them on my plates. So I reached out to some of the people here at VIMS and the University of Maryland, and the way I told them, I was just like, I assume they're anemones. However, I don't know, but if this helps, they're very sticky to the touch, which they are. And they were anemones. And I also really like, well, I don't like the sea squirts, but it was impressive to see how many of them were in some of the bags. They would just cover everything, which was insane. I'd never seen anything like that before. Yeah. And this one's not even that bad. Oh, yeah.
Great talk. That was really interesting. So you used kind of two different methods, the method of looking at everything under the microscope and identifying everything, and then also the ImageJ that looked at percent cover. And so a lot of us who look at invertebrate community dynamics are trying to find quicker ways to, assess community structure. And so I'm just curious, the difference in time it took you to identify things under the scope versus doing the processing with ImageJ. would be a time saver to do things with photos instead of the microscope?
So when I used ImageJ, I didn't use it specifically to identify taxa. I just used it to identify like percent cover or percent open, sorry. So I think it would be difficult using pictures, especially through ImageJ, because I don't know if you've ever used ImageJ before, but you know, it's very, very time consuming. And that's why some of my pictures, sorry, like a lot of my pictures have that blue backing because it made it so much easier to process them through ImageJ. Because if you have any sort of shading or anything on that background, it will not read it how you want it to. So the processing those images took a few days, a long time.
And that's just for percent cover. So
And that's just for percent cover.
do you
open.
think you would be able to identify species with the photos, or is that out of the question?
I'm sure there might be ways through ImageJ. I don't know everything about it, but probably not. So I think it would be for the best to just go through the plates and look everything under the microscope.
There's no replacement for looking under the microscope.
Not really. It's better that way. And I was told by Jennifer Dreyer that in order to identify species more accurately, it's better to look at them alive in their environment. So doing this was more difficult, even though, but so I was lucky that I didn't have a lot to look at. Yeah.
Great, thanks.
Hi, great talk. I wanted to ask, did any of the biofouling organisms seem to show up more often than any others?
Yes. So the anemones, there weren't a lot of them, like they wouldn't cover entire plates or anything, but they were on most of the plates, as well as the bryozoans. I mean, you can see from my other pictures, they're there. They're everywhere. They're just on everything. But and the algae as well. There was a lot of algae there. Nothing too exciting, but yeah. The sea squirts surprisingly did not show up in a lot of my plates except like 2. But when looking at the actual bags, sea squirts were there everywhere. Yeah.
Then I think we are now able to go on to the next presenter. Thank you so much, Sophie.
So our next presenter is going to be virtually because Grace Kirkpatrick had the opportunity to go out with her mentor, Dong Lai Gong, and experience work with the glider out in the field today. So she did a pre-recorded presentation, and it might just take us a minute to get everything set up. But Stephanie's our tech guru, and of course we have Will here to help out. So hopefully it'll go seamlessly. But I'll pass it back over to Sarah for the brief introduction, if we're ready. Give us 1 minute.
Thank you so much for being here. You're watching my presentation pre-recorded today because I am currently out on a boat deploying a glider. So I was working with Dr. Donglai Gong this summer on quantifying errors within data-assimilative global ocean models using glider data from the Mid-Atlantic Bight Slope Sea. So for some background, this project was undertaken with data gathered in collaboration with NOAA's Hurricane Glider project. This initiative is informed by the fact that over the past 20 years, hurricane trajectory forecasting has significantly outpaced hurricane intensity forecasting. And this general lack of skill in hurricane forecasting costs both money and lives. Hurricanes have cost the US over $1 trillion in damages since 1980 and have caused over 10,000 deaths. Recently, hurricane intensity forecasts have been shown to improve when they integrate accurate real-time data about the upper layers of the ocean over which they're traveling. One place that this data can come from is from ocean models. But these models in particular are not perfect. In the Atlantic Ocean, the Gulf Stream is one major source of uncertainty. It's a narrow, strong, warm, and salty northwards current that stretches up the East Coast and eventually starts to meander off towards Europe, transporting warm water and heat polewards. But because cold water can suppress hurricane intensity and warm water can increase hurricane intensity, accurately representing the Gulf Stream is important to hurricane forecasting. Unfortunately, ocean models have historically struggled to fully resolve the extent and dynamics of the Gulf Stream and the eddies that spin off of it, including the temperature gradients that make it up. One of the ways in which the accuracy of ocean models can be improved is by processing and assimilating real-time observational data from vehicles like gliders and sailplanes, and algorithmically modifying their predictions accordingly. Gliders, which my lab works with, are particularly useful for this kind of data assimilation. They can be safely deployed for months at a time with minimal supervision, but they can be steered, unlike floats, and they can safely gather data during extreme weather events. They can also gather three-dimensional data because they can dip down deep into the ocean. The downsides are that occasionally sharks do like to nibble on them. On the right, you can see a graphic of how a glider typically moves through the water. Gliders have a characteristic sawtooth pattern as they move through the water. At the surface, they transmit position coordinates and some data back to land and are then instructed to deflate their buoyancy engine, which causes them to sink downwards and travel forwards in accordance with their heading and the currents that they're traveling in. When it hits the bottom of its cast or its downward descent, it inflates the buoyancy engine, causing its density to decrease, which leads it to ascend through the water column back to the surface, where it again transmits data. The picture to the right is of a glider in the lab, not one of the gliders that recorded the data sets I'm using, but a very similar one, just so you can get a sense of what they look like. Our objective for this summer was to investigate how the output of one globally constrained ocean model that assimilates glider data, RTOFS East, compares to the observational data that was collected by two gliders, the Navy Glider 657 and the Navy Glider 738, which were deployed from about October to February of last year. We were broadly interested to find out how accurate the model output was to the data the gliders collected, and thus how well the model was assimilating the glider data it had been provided. We anticipated that there might be some differences because the slopes sea and the shelf break are dynamically complicated areas, and the Gulf Stream also poses challenges for modeling. To begin with, all of this analysis was conducted with Julia, which is an open source high performance programming language. Here you can see two maps. The one on your left is the path that NG657 charted over the course of its deployment, while the right one shows the path that NG738 charted. Note that NG738 covered a much larger geographic area than did NG657, as you can see in the top image. In the larger box bounded in green represents the range of NG738, while the smaller blue bounded box represents the range of NG657. This data was a little difficult to analyze because the temperature and salinity field of the Gulf Stream changes with time, and the glider that is recording this changing field is also moving in position, as well as in 3 dimensions. Any point on any of these trajectories could realistically represent a time when the glider was actually 1,000 meters below the surface. So I'm going to present some depth transects and some animations to kind of look at this data from a bunch of different angles, but all of them are necessary to get kind of an accurate, bigger picture understanding of how accurate this glider data is to the model outputs. The other complication to this, to my data analysis, was that the glider provides much more finely gridded data than the RTOFS model does. RTOFS is a 1/12th degree resolution model, which at these latitudes kind of works out to about a 4 mile by 4 mile grid and provides forecasts every 6 hours. And a glider moves with the currents, so they can take multiple measurements within the spatial and temporal boundaries of a single model output grid, sometimes up to over 10. So to achieve a glider output grid that had the same resolution as the model output grid, I had to bin all of the glider data by the temporal and spatial boundaries of the model box they were taken in, and then average those bin data and use them to create these transects. All right, so potential temperature. So when we look at these graphs, potential temperature is on top and practical salinity is on the bottom. For each graph, depth is on the y-axis and time is on the x-axis. The potential temperature color bar has higher temperatures represented in orange and lower temperatures in purple, whereas the practical salinity color bar has high salinity values in yellow and low salinity values in green. I'll talk about these more a little bit later, but I just wanted to point out the big light colored band in the middle of these visualizations. That is the Gulf Stream signal. I also want to point out that even visually, these transects seem to be reasonably similar. Moving on to NG738. Again, we have time on the x-axis and depth on the y-axis. But we can see that the Gulf Stream pattern here is a little more complicated, and it doesn't match up quite as well between the model and the glider, especially past the timestamp corresponding with January 1st, 2022, which is about 3/4 of the way through the transect to the right. We can understand these transects better by calculating some overall statistics of model skill. I chose to use the root mean square error between these models and the bin glider observations. In an absolute sense, the errors associated with the model glider comparison are not negligible, but are fairly low. But although the absolute errors are pretty low, the errors associated with the practical salinity along NG738's trajectory are 32.2% higher than those for NG657. And the errors associated with potential temperature along NG738's trajectory are 48.6% higher than NG657's. To understand why, it helps to consider these series of transects, which represents just the raw difference in value between the model and the average bin glider data along each glider's trajectory. So you can see that for NG657, with some small exceptions, the difference graph reflects the visual similarity of the original transects. It certainly isn't perfect, but it's relatively consistent with the low level of quantitative error. But in NG738, this is on the right, this is not quite the case. There's an area of pretty significant divergence between roughly January 1st and January 18th. You can see with the color bar, areas in red represent areas where the model overestimated what the glider said, and areas in blue are where the model underestimated it. And between January 1st and January 18th, the glider moves through an area where the model seriously underestimates the actual temperature and salinity of the water, and immediately moves into a region where the model seriously overestimates the temperature and salinity of the water. And based on the sheer magnitude of these temperature and salinity differences, this has led us to believe that during this time period and in this specific geographic region, the model was not correctly resolving the extent and the kind of landward boundary of the Gulf Stream. So to kind of zoom in a little further on this issue, to the left, we have an animation that I'll be playing in a second of the potential temperature at the 200-meter depth with the glider's trajectory and position overlaid on it. On the x-axis, we have longitude, on the y-axis is latitude, and the yellow end of the color bar represents higher temperatures, while the purple end represents cooler temperatures. Since one way to define the Gulf Stream is as the area in the ocean where the 200-meter depth exceeds 15 degrees Celsius, I've set up the color bar so that the distinction between temperatures characteristic of the Gulf Stream and those that aren't are pretty visually distinct. On the right, on the right top, we have a zoomed in segment of the NG738 difference graph from the previous slide for easy reference. As we play the animation, you can see that during the period of January 7th to 10th, the glider is in an area where the model records it as being decidedly outside of the Gulf Stream. But when you look at January 7th to 10th on the transect, it records temperatures characteristic of the Gulf Stream. And we estimate that during this period, the model and glider estimates differ spatially by up to 12 to 16 miles. But between January 10th and 16th, you can see that the model records the glider as being pretty far inside of the Gulf Stream, but the transect records that the model significantly overestimated the temperature of the ocean at this place at this time. And during this period, we estimate that the model and glider estimates differ by up to 30 miles in some places, which is fairly significant considering the narrowness of the Gulf Stream. I do want to note that these estimates are very rough. And so the specific numbers should perhaps be taken less seriously than the implication, which is that RTOPS both underestimates and overestimates the extent of the Gulf Stream in a small geographic region, which hints at an underlying problem in how RTOPS trusts and assimilates glider data. So to summarize, overall, both NG657 and NG738 reported low quantitative errors, and both did reasonably well in areas that are decidedly outside of and decidedly inside of the Gulf Stream, and struggled mostly where the boundary between the Gulf Stream and kind of the normal ocean is undetermined. However, the model failed to properly resolve the landward edge of the Gulf Stream in at least one instance, sometimes by up to 30 miles, which indicates that the model is not sufficiently trusting of the observational data that these gliders are providing it, and it isn't assimilating it correctly. Areas of further research for us might include investigating whether the mean position of the Gulf Stream, and not just its landward edge, are not being properly resolved by these models because of errors in data assimilation. It might also involve comparing these results to those of other models, like the regionally constrained DOPIO or GOFS, the Navy's ocean model, to determine whether this difficulty is in kind of a dynamics problem, or whether it's because of RTOP's specific data assimilation schema, which differs between ocean models. So thank you for everybody who contributed time, ideas, or materials for this project. Thank you all for being here, and a special thank you to my mentor and to my fellow REU students, as well as Stephanie Pert, Dr. Grace Massey, and Dr. Rochelle Seitz for organizing this program. If you have any questions, I would be glad to answer them. If I don't make it back in time for Q&A, you can contact me at the email address on this screen, and I would be more than happy to talk to you about my project. Thank you so much.
Yeah, so that was Grace, and her email is visible on the screen in case you have any questions. You can send them over to her. But yeah, and so we are running a few minutes ahead of schedule. I didn't know if we just wanted to wait a couple minutes. Cool. So as we set up for the next presentation, we're just going to need a few more minutes, but we'll be back around 11:10.
Hi, everyone. So if you can start getting settled, we're going to start our next presentation moment and well, right now.
Alrighty, everyone. So after that short break, we are now going to watch Grace Levins from Union College discuss sea marshes. Nope, salt marshes. There we go.
All right. Hello, everyone. As Sarah just mentioned, I'm Grace Levins, and today we'll be talking to you about private levees and their impact on marsh migration. So to start, I wanted to do a little overview of a natural transgressing marsh. On our left, you can see there's typically a tidal body of water here, transition into low marsh plants, high marsh shrubs, and uplands. The brown section here indicates the depth to organic matter, and this generally decreases as you go into your uplands. A marsh can exist like this essentially forever, but with rising sea levels, marshes are migrating inland into this upland areas to avoid drowning. This can also continue until there are barriers in the way of the marshes. So that would prevent the marsh migration, and then it would drown in that situation. Migration becomes an issue when it's starting to migrate onto private property and or agricultural land. In this setting, landowners will typically put in a levee between the high marsh and shrub boundary to prevent flooding onto their personal land. I also just wanted to briefly go over what is a levee for those of you who might not know. In both of these images, the base of the levee is indicated by the yellow dashed line. You can see in the picture at the right, they can be pretty substantial. That's my grad student on the top of the levee, and I'm taking the picture at the base. There's recorded levees of being 6 feet tall, so there's a wide range in levee height. It's generally a function of tidal range. So levees are typically made up of sediment from the surrounding area, typically on the landward side, and they support the growth of salt-tolerant trees, as you can see in these images here, like red cedars and loblolly pines. For my project specifically, like I had mentioned, the driving problem is sea level rise, and that can be seen in the effects of flooding, saltwater intrusion, and rising water tables. Our study sites are in Somerset County, Maryland, box labeled A, and Accomack County, Virginia, box labeled B. And one of the reasons, or some of the reasons we're studying this marsh migration are because of the socioeconomic impacts, like lower crop yield, reduced property values, and land loss as marshes are migrating onto private property. It should also be noted that this project is an extension of a prior project looking at the effectiveness of levees in the Gloucester County area. And in a accumulation of our data and the project before, we found that levees have existed in the Chesapeake for centuries. So once we got to each of our sites, we chose the sites based on levee condition. So there are some on active farmlands, some on restored marshland. At each of our sites, we set up 4 parallel transects, which I will show a map of later. And you can see an example transect in the marsh side in this image here. As we were setting up the transects, we took an RTK GPS. This is me with the rover here. And the RTK GPS provides high resolution location elevation data. We took elevation points every 5 meters along the transects. And while we were doing that, we were also recording marsh vegetation, as that's another metric of levee effectiveness. Once that part was complete, we went back over the transects on both the seaward and the landward side and took soil samples and cores to later process in the lab. So once everything was back, I did 3 main analyses. The first I started was with soil moisture. I transitioned the sediment from plastic bags that we're using in the field into these metal tins here. I took a wet weight, dried them at about 100 degrees Celsius, and then reweighed them to find the difference. And then I ground each sample and rebagged them with a correct label for further processing. My next lab procedure was to test for salinity. This was a bit longer. I weighed out about 10 grams of sediment into these plastic jars here. I saturated the soil with 5 times the amount of deionized water, shook the jars 4 times over 2 hours, and then let rest overnight. The next day I would come in and take a new jar, put a coffee filter on, and pour the saturated soil over it to drain out the liquid. And then I took the salinity of that liquid. The last test I did was loss in ignition, otherwise known as LOI. This was done by weighing out about 5 grams of sediment into these metal tins here, muffling the tins at about 500 degrees Celsius to burn off any residual carbon. This LOI, it just gets you your percent organic matter, which is another metric of marsh age. Just a brief little overview of all the work that we did. We spent a lot of time in the field, but it was super essential to collect all of our samples. In the Virginia sites, we did end up collecting about 169 samples and 16 cores. Maryland, we've only done 2 out of the 3 sites. So this is as of right now, but I'll talk about that in a bit. And so, yeah, this was mostly how I learned everything, was just being out in the field. That was really great. So these are our Virginia sites. BC stands for Bundick's Corner, BL is Bundick's Levee, and CL is a cattle levee. You can see how close the Bundick's Corner and Bundick's Levee are. And these are active farm sites, and the cattle levee is a restored marsh site. So these are the transects that we ran. You can see how close these are. This corner is this corner up in the top. They're just separated by this creek here. And the cattle levee, this side is our landward side, and this is the natural marsh. So these are the elevation profiles we took with RTK GPS. The orange line here indicates the elevation. The black line is depth of parent material. So this orange space is the depth that we found at each elevation point. And the dashed line indicates highest astronomical tide. We have distance on the X and elevation on the Y. So our Bundick's Levee and Bundick's Corner, because they were never marsh on the landward side, you can see they have a higher elevation. Just the leftward side is the seaward. This is the peak is the levee and the right is the landward. So the 2 agricultural sites you can see are higher in elevation on the right. And at our cattle levee site, because the landward side was marsh at one point that was drained and now is being restored again, it is at lower elevation, potentially because of subsidence or compaction from when it was drained from its original marsh state. Here are our lab results. So starting in this top left graph, we see site along the x-axis and average depth of parent material along the y. There's a higher depth of organic matter on the seaward side because of continued saturation and anaerobic conditions, which slows organic matter decomposition and allows for accumulation of organic matter. And we see the reverse, lower percentages on or depths on the landward side just because of the aerobic conditions. Our top right graph is of salinity. Again, sites on the X, average soil salinity on the Y. We're seeing higher salinity values on the seaward side because of tidal inundation. And there's this blip kind of on the Bundick's Levee site. When we were out there, we noticed that there was a breach in the levee, so it could be a result of that. Our bottom left graph is of the LOI value, so loss on ignition percents. You're seeing that it's higher on the seaward side just because LOI increases with marsh age and there's higher organic material over there. And it should be lower on the landward side just because there's less organic matter. Our final graph in the bottom right is for soil moisture, and it should be higher on the seaward side just because there's continued inundation. It's a marsh. And overall, we're seeing that the two farm sites, BL and BC, are giving the data that we want to see. So at each of our sites, we found that we were there for long enough to see both low and high tide. So when we were there at high tide, there was no tidal inundation on the landward side of the levee, proving that it is effective in preventing tidal inundation. But there is formation of new marsh vegetation, as you can see in this top image here. These are Phragmites, which are common marsh plant. And we could also see it on the other side of these shrubs here on the Bundix levee. At the Cattle levee, we saw decreased values in depth organic matter and LOI on the landward side. This means that at one point, the reclamation of the land was effective when the people were first transitioning it from marsh to cattle grazing field. If the levels had been the same, then that would have meant that it had been marsh for that whole time and there was no conversion. So overall, we're seeing that the levees are effective in preventing tidal inundation, but additional technology is needed because at some of the sites, we were noticing that there could have been pooling of water on the landward side. So if there was something like a tide gate installed, it could drain the water on the landward side and you wouldn't get this marsh vegetation growth. So for future work, my first step is to complete fieldwork in Maryland. So these are our sites here from Maryland. We've been to this star and this star, and I will be visiting this one right here. These two are our active farmland sites, and this is an abandoned levee. So we'll be able to compare the statistics again once we get all of our data. I also am going to process the rest of our soil samples from Maryland sites and process the cores from the Virginia and Maryland sites for bulk density. This is an example core here. You can see this is how we'd measure the depth to parent material. And I'm also going to complete statistical analyses on all of our data. So I want to give some acknowledgements. First to Dr. Matt Kerwin for allowing me to work in his lab and for providing me with continued support as I get through this project and continue into my thesis work. Grace Molino for showing me the ropes of salt marshes on all of our fieldwork expeditions and answering my endless stream of questions. The Kerwin Lab for continued help in the field and the lab. All involved in running the REU program here for providing me this incredible opportunity. My mom, Martha Petrallo, for helping with plant identification when we were out in the field, and Cole Miller for your assistance during fieldwork, even when we're tromping through the densest of vegetation. So I'm now going to leave you with a picture that we took at the Cattle levee with our drone. You can see this is our levee here with the trees. This is the landward side, and this is the seaward side. And I'm now open to any questions.
So if now anyone has any questions, Stephanie will be around.
Hey, great talk. You mentioned something about needing additional technology to be able to tell whether there was any pooling on the other side. So what do you mean by that? What type of technology would you need?
Yeah, so at one of the sites in Maryland, we actually saw that the landowner had installed tide gates to be able to drain the water from the field that he was growing. He was growing millet on. So it's just a way of controlling the amount of water on the landward side. So because you can see, here, I'll go back to my elevation profiles. You can see that there's a bit of a slope here from this is like the middle of the agricultural field, not even the middle, that's massive fields, but there's a slope here. So when it rains or if there's any water on this, it tends to pool in these areas. And that's probably promoting the marsh growth. So if there was a way to get rid of this water and drain it into the marsh without, or like a way that it would only open into the marsh side and not allow any salt water in. So that's what a lot of tide gates, their function is. That would probably help keep the marsh plants out of the landward side.
Thank you.
So you mentioned that there was a breach in one of the levees. How often does that happen? Like, is that common? And how do like landowners fix it?
So the breach that we saw, we hypothesized that it was actually cut by the landowners. At this point, I don't know why it was cut. Typically, if somebody's going to breach their levee, it's to restore the marsh side. So like at the cattle levee, there was a breach in the levee to restore the marsh. Generally, a levee shouldn't have a breach in it because then it decreases its effectiveness. So we only really saw it at this Bundix levee site. But I think if you're going to restore it, it would just be adding more sediments and building that dune back up, basically.
Yeah, I had a similar question. Like for during storm events or like high energy, like weather events, is there any sort of upkeep that you have to do for these levels? Like is erosion a thing, or like over what time scales would you have to worry about? Okay, I've lost a lot of my levee. I have to think about starting to rebuild it.
Yeah, so I think it depends on the levees we're looking at. So the ones in the farm sites, the landowners they're actively using them. So they're checking on them, they're maintaining them, they're making sure that they're doing what they're supposed to. And like I mentioned, if there's a breach, like there's just general upkeep with them and making sure they're at the height that's going to work for, because this dash line here is the highest astronomical tide. So if your levee is higher than that, then essentially it should be effective. Storm events obviously are an anomaly, which hopefully I'll be studying with my thesis work. But I think it's just general upkeep. If you want your levee to work, you have to put in the work to make sure it's working.
Hi, Grace. Great talk. Okay, I have two questions that are pretty dissimilar, but I'll ask them both at the same time. The first is how, once you analyze the vegetation data, so I know y'all were recording the different types of vegetation as you walked your transects, how do you see that that analysis playing into the data that you already have? Like how do you expect to incorporate that into your analyses? And then my second question, these elevation profiles are really cool, and there's a lot that y'all have included in these figures. And I'm interested, these levees are kind of situated right at the brink between your land that will be flooded every day and the land that won't. And so I'm interested if you have thoughts about, why these levees were built at this specific distance, and why they maybe weren't built like closer to the left, I guess, to try to preserve more land. And then how do you assume that, like, you're assuming that the levee is responsible for some of the effects that you see on the landward side, but how do you kind of parse out, like, what is the levee responsible for versus just what is that higher elevation land responsible for, and any of the changes or differences you're seeing on both sides of the levee?
Yeah, so to start with the vegetation data, I just quite honestly didn't have time to add them into the elevation profiles. When I write my paper, I'll have the difference in vegetation here at the top, so we can see how it's transitioning as you're going from the seaward side into the landward side, and if we're seeing any of the marsh vegetation on the landward side. So that'll be another next step. with regards to levee placement, that might just be something that we have to talk to landowners about. That's not something that I know to my knowledge of why they might have placed it where they did. It might have just been the edge of their land and where the marsh has existed, because at these properties, like this side was never marsh, so they probably placed it at the marsh boundary to prevent any further migration onto their property. And then remind me of your third.
Yeah, just a continuation of that.
Yeah.
How do you parse out what the levee is responsible for, what differences the levee is
Right.
responsible for versus the higher elevation?
So I think that'll be like at this point in my research. I'm not sure if I can completely answer that one. But I think what we've talked about is just that, well, we haven't really talked about much of that, but that'll just be something that I'll have to look into further, and I can get back to you on that.
Alrighty, so everyone. Then next up, we have Anissa Sellers from Virginia State University, and she will be talking to us about microplastics.
Okay, hello, my name's Anissa, and this summer I work with BK Song and my amazing mentor Mara Walters, looking at the effects of different microplastics on oyster larvae survival and microbiomes. Okay, so we were looking at the Eastern oyster. It has a very important ecological and economical significance here in Virginia, especially here on the East Coast. So for ecological reasons, it's important because not only does it help filter the waterways as filter feeders, they also act as the basis of various communities and species because they form these oyster reefs. Yeah, and the economical, again, I'm gonna take a little minute, but I got this. Okay, so I'm gonna start a little bit. It's okay, work with me now. We're gonna get it done. So I'm gonna backtrack, but it's okay. So first of all, ecological importance, I briefly mentioned that. You know, they filter waterways, they form the basis as oyster reefs. Economical, Virginia is one of the top producers of aquacultured oysters. This is important because not only does it prevent the depletion of natural populations, it allows for us to support and maintain or meet the demands of the human demand for the seafood. However, as the industry grows, there are some different changes that the hatchery needs to make to ensure the growth and survival of the oysters themselves to produce the yields that they want. And with those, there's an emerging concern of actual water quality, what's meeting these oysters, because like I mentioned before, they're filter feeders. Whatever's in their environment directly impacts them. It comes in contact with them. And so water pollution is a huge thing. Whatever's in the water, it may have the potential to affect them. So one of the biggest polluters that we see in the environment, particularly in marine environments, is plastics. So plastics are used in our everyday lives. We have them in clothes, such as right here with the nylon fabrics. We have them as containers. Down here with the HDPE, which stands for high density polyethylene. So in containers, storage, packaging, things like that. However, the main issue with this type of pollution is that they stay in the environment for hundreds of years, like a long time. That's because they do not biodegrade. They only break down and get smaller and smaller. And that's when we get to microplastics. So microplastics can be defined as anything or any plastic, to be more specific, any plastic that is smaller than the eraser of a pencil. And there are two types of microplastics. You have primary. primary microplastics are basically microplastics that are already broken down for consumer use. So if you think of facial exfoliants, those little micro beads are microplastics. In addition, secondary, it's more common, and they refer to the larger plastics that get broken down into smaller pieces. So this is a growing concern in that if they get small enough, they may be able to infiltrate the filtration systems that oyster hatcheries have in place, which would not be good if they come, well, it may not be good if they come in contact with the oyster larvae that they're trying to grow. However, I would like to stress that not all microplastics are going to be petroleum-based or fossil fuel-based, and not all of them may have a negative effect. Okay, so I'm gonna switch it up a little bit and I'm gonna explain it's gonna come all together in the end. So a microbiome, just for some context, is a collection of microbes that live within or on your body. But it's not just human bodies, it's also various organisms that have them. So previously, there have been some studies that have proven successful with the supplementing the use of probiotics as a food supplement to oysters, as in addition to various other aquaculture shellfish that have proven to actually increase or be beneficial to the survival rates of these organisms living in this artificial setting. So probiotics are alive, non-pathogenic bacteria that add to an already established microbiome. And below we have some examples of probiotics that we've seen used. We'll be looking at a kind of novel technique is the use of prebiotics on these aquaculture shellfish or bivalves. And prebiotics are a non-digestible food supplement that induces the growth of a microbiome. So instead of adding to something, it's basically making it stronger. You can also think of it as a vitamin. And here are some of the examples. So I want you to pay special attention to the polybeta hydroxybutyrate, which stands for PHB. And PHB is a bio-derived microplastic produced by bacteria that acts as a prebiotic. So basically what I'm saying is we're gonna feed a vitamin to oyster larvae that's basically a microplastic. Okay, so the objectives of our project are to look at the effects of these microplastics on the oyster larvae survival and growth. In addition to, we're going to compare the microbiome to see how these microbiomes, if they had any effect on the actual microbiome, if the microplastics had any effect on the microbiomes themselves, and compare and contrast and see any differences. Okay, so our overall methods, we started with the feeding experiment. We had 5 overall treatments of the control, 2 different concentrations of the PHB, the nylon and HTP. So I have some pictures to kind of show you what they look like. After the initial feeding experiment, we then did DNA extraction and then DNA sequencing to kind of see what we had there. So experimental setup. So the feeding portion at least took place over 9 days. We worked in the ABC hatchery, or the VIMS hatchery that we have here. For each of the 5 treatments that we had, we did 3 replicates. So in the end, we had about 15 jars total. For each of those jars, we received 10,000 larvae from the hatchery to place in them. So our kind of daily life would be to check on the larvae. Water changes occurred every other day. And with those, we would filter them out and then look at them under a microscope to assess them for health and movement. And we also count them. So up here is an example of what they would look like when they were alive and happy. Okay. So at the initial feeding section, that's when we did the DNA extraction. We used a DNA extraction kit to extract DNA from both the oyster larvae themselves and the water. So we did the both to see if there's a contrast in microbial communities to see if the water may have an influence on the oysters and so on and so forth. So in the picture with our little frog, Frederick, I love him. This is what we use to kind of run the PCR. PCR stands for polymerase chain reaction, and this is a thermocycler, this machine. But yeah, it was used to basically amplify the DNA that encodes for the 16R RNA gene. And that is basically a gene that's found only in prokaryotes, which is bacteria, and what we're looking at for microorganisms. Then it was then sequenced, and then we used the data-to and phyloseq packages in our program for bioinformatic analysis. And basically what that means is we looked at all those sequences, kind of sorted them and took out any errors or chimeras. Chimeras being any sequences are accidentally merged together and are incorrect. Okay, so overall survival, not the best. The highest was a little bit less than 13%, and that was the control. So in addition to the overall kind of lower survival rate than we expected, we also saw a decrease in the growth. So this is kind of the pattern that they should be growing in from day 1 to 10. So we expect them to be around the size of the day 10. However, that did not occur. They were more between the sizes right here. So about 3-4 days old when they should have been towards the size of the 10-day-old larvae. To calculate the survival, we did the counting of the live larval divided by the initial larvae that we received, the 10,000, and then multiply it by 100 to get that percent. And I also forgot to mention, but if you look at the graph on the x-axis, we have the treatment, and on the y, we have the survival percentages that continues for the next couple of slides. Okay, so we ended up splitting that initial chart of overall growth, or not overall growth, but overall survival into 2 intervals. Because it was so low, we want to see if there are any patterns in between each section. So we looked at the interval from day 2 to day 4 larvae. And if you look kind of closely, you see that the petroleum-based microplastic nylon was kind of high, which is kind of concerning. We would have thought that the survival would have been low with the petroleum-based if they had, actually taken them in. But there are also various other things that could account for this, because the week of the experiment, we had high temperatures. We had a few issues with the aeration and various things that I'll go into a bit later. But I also want you to take special note of the 0.1 milligram per liter PHB. It is the lower concentration of PHB. However, it had a greater survival compared to the higher dosage of the PHB. So it can be inferred that they may prefer a lower concentration, but we'll get into that a bit later. Okay, so for the latter half of the experiment from day 4 to 9, you still see an overall decrease. However, that it keeps that same trend of the lower concentration of PHB being higher or greater than the higher concentration. I also wanted to note out that if you look at the HDPE, which stands for the high density polyethylene, you can look in the picture and see that it kind of aggregated on top of the water, at least in this solution that we tried to make. And so in the actual hatchery setting within the jars themselves, it didn't really disperse itself throughout the water. So we're thinking that maybe the larvae didn't get into much contact with them, and therefore it had a bit higher survival rate compared to the actual nylon. And then here's the overall comparison between the two. So overall survival rates were below 13%. But if you look at the comparison, you can kind of see a trend. Take note that the scales are different. This is from 100 to 20. This was from 30 to 0. And so you see a switch in trends from HDPE and nylon. Nylon was higher and HDPE was lower, and then it kind of switches off down here. But there's that same trend of the lower concentration of PHB being higher than the 1 milligram per liter PHB. Okay, so this graph is a principal coordination plot, or PCOA. It looks very intimidating, but I can make it, a bit more manageable. Give me some moment. So the different axes are going to explain the amount of variation, or percent of variation. So this first axis is a 49.1% variation, and then this axis is a 11% variation. The different points on the actual graph are the different samples. So the circles represent the water samples that we took, and the triangles represent the larvae samples that we took. The different colors are the different treatments. The one big thing I want you to take away is the different clusters. So you see how all the water is kind of clustered together, showing that they're more closely related and a bit more distinct compared to the overall larvae, which are kind of clustered to the side over here. But you can also see within the larvae, there are a few clusters as well. Like if you look at the 1 milligram PHB, which is this green triangle, they're kind of close together, which shows some similarities within some of the treatments, but not too many. Okay, so for the microbiomes and the oyster larvae themselves, it was kind of all over the place, the same type of pattern, the same type of graph. However, the different shapes represent the actual different replicates. So replicate A, B, and C. Since there's no definite pattern, you can see that there is a good amount of, diversity, but the treatments don't have a specific effect that you can see. So there is no obvious effect on the microbiomes. Like you can kind of see a bit of a cluster here, but it's not very significant. And then finally, we looked at the alpha diversity of the larvae. And the alpha diversity is just referring to the overall amount of diversity that we found, like how many species were there. So for the control and nylon, you can see that they had a pretty high amount of species diversity. So they had about around 100 species within those treatments. Overall, it was pretty high, but yeah. To the next. Okay, so to conclude, survival of the larvae. So the oyster larvae may just prefer a lower pH concentration, as shown with that same trend. But there are also various possible explanations for the high mortality, things that we can work out later on. Particularly the high temperatures. While working in the ABC hatchery, it was kind of hard to control the temperatures, and that week was particularly high temperatures, like in the hundreds, and all we had was like a fan to kind of cool the jars down. And also aeration was a big issue. Sometimes aeration would like kind of cut off because we were making sure the aeration was low. Since it was already kind of low, it would just shut off sometimes, and we would come in and they wouldn't have air, and various other explanations that I'll get into at the very end. For the microbiomes of the larvae, there was no specific pattern within the larvae themselves. They were kind of dispersed all over the place, as shown with the previous graph. However, the larvae had very distinct microbiomes compared to the microbial communities in the water, showing that their microbiomes are selective. They aren't very influenced by the water. They kind of specify what they have. And there was a pretty good diversity within the highest diversity were in the control and nylon, but overall, you saw a pretty good diversity of different species within each of the treatments. For future directions, some advice that we had gotten from both the directors of ABC and ACUF, the hatcheries, were the possible use of older larvae. Maybe that the day 2 larvae that we started off with were too young and too, stressed to, survive and grow. If they're older, they may be able to handle that stress better. To work in a better and more stable environment, better control the temperatures, aeration, and pH and things like that. I also wanted to continue this research or see if others would continue and see if the oysters actually ingested any of the microplastics and maybe how that could affect us. Because if they're ingesting them and actually surviving and growing up, as humans who eat them, how will that affect us? And a better closely follow hatchery procedures. What I mean by that is we had a bit of a different setup. We use splash jars instead of the plastic jars that they use to prevent any plastic contamination, but maybe better following how they do their procedures, as well as looking into the volume of the oysters that we used, because the volume may have have to do with the high mortality rates as well. But yeah, I also wanted to say thank you to the NSF for funding me, BK's lab for accepting with open arms and being so helpful, particularly Mara, because Lord knows I was very dramatic this entire summer, every day at work. Gail Scott for sequencing our DNA for us, Jessica, Amanda from ABC, and Lauren from ACUP for helping with the larvae. Everyone from REU, especially the coordinators, they're amazing and so helpful, and the interns who are the absolute best. Any questions? This is me and my son, Jimothy, my little oyster larvae baby.
Great. And because of time, we only have time for like 2 questions, maybe.
Okay.
Great talk, Nisa. I was curious if the scope of your project included other prebiotics or microplastics, and if so, what caused you to eliminate them from your final design?
What caused me to what? I'm so sorry.
I was curious if your experiment included other prebiotics or microplastics, and if it did, what caused you to eliminate them from your final design?
No. So for what my experiment is a continuation of Mara's initial one. And so I know initially they had a similar setup with the petroleum-based microplastics and then the PHB. The only difference between those two is that I use a lower concentration overall. They use like 10 milligrams per liter PHP, and I just use like 1 and 0.1. But as for other microplastics, I know that I chose the nylon HDPE because they're more prevalent in as pollution. I kind of referenced them in the very beginning. They're kind of, you know, a bit more prevalent, especially as microplastics in marine environments. They have been shown to be common. So that's something I could look forward to in the future, looking into different types of microplastics, things like that. Thank you for your question.
Thank you for answering.
First off, great talk. I'm not really well versed in like larvae stuff. So I believe you said you had 10,000.
Yeah.
And then you got like the survivorship based off live versus initial.
Mhm.
How did you get the live count? Like, did you individually count all 10,000?
No, we did not individually count. So we received the larvae count from the people in the hatchery. So what they would do is they would get a sample. Then within that sample, they would get another sample, kind of do math, and then kind of estimate the amount. And then they would give us kind of a measurement to take out. So if they give us like 100 milliliters of like water and they knew the concentration of how many larvae were in there, then they would tell us like take out 3 milliliters, and that'll be like around 10,000 larvae. And so that's what we would do. And then for the counting, we would take, you know, when we filter them out, we would kind of condense them or concentrate them as well into like a similar setup, like maybe fill it up to like 100 milliliters, you know, of water, and then take a sample of that, look at them under the microscope. So to determine if they're alive, we would look at first for movement, if they're swimming around. Then we would look at gut, you know, if they weren't swimming around, then see if they had active gut. If they did not, and it was empty, they were dead. If they're happy and swimming, they're alive, which is good. And then for the counting of those, similar, however, we use a knockout solution to kind of put them to sleep so they stop running around. Look at them again, count them live ones, and then do math and figure out how many were alive, how many, well, we didn't really take into account how many were dead, but at least how many were alive. Any other questions? Thank you.
Alrighty, thank you, Anissa. And for the last session of our, the last presentation of our morning session is Lindsay or Smitty Smith, and she is from William and Mary. And yeah.
So thank you for that introduction. Like Sarah said, my name is Smitty, and I worked with Dr. Mark Brush and Stephanie Peart, and I'm excited to talk to you all today about different methods for studying phytoplankton community metabolism. So I'm going to start by describing to you all how important phytoplankton are to ecosystems. So they provide 50% of global net primary production globally, and this includes being the dominant source of primary production in most estuarine ecosystems. Similarly, they provide organic carbon to the food web and support the biogeochemical cycling of nutrients throughout their environments. So in order to study phytoplankton community metabolism, we look at the changes in oxygen over time. This is because, as you can see in the photosynthesis equation at the bottom of the screen, oxygen is a product of photosynthesis. So the traditional method we use to study these changes in oxygen over time are to use the light dark bottle method, in which we put we incubate water samples in in-situ temperatures in a light box, which can be seen up here, where they are kept at different light levels and changes in oxygen are recorded. This method is both time and labor intensive and requires a high volume of water. So for this experiment, we were also comparing the light dark bottle method to the Hanstech Oxytherm Plus photosynthesis, which is this machine right here. And I will be referring to it as the Oxytherm for the rest of this presentation, which is a temperature and light controlled chamber that continuously monitors fine scale changes in oxygen concentrations. This machine or chamber requires a smaller volume of water and has been used to study aquatic species, but has yet been used to examine estuarine phytoplankton community metabolism. And so our hopes were that this machine would be less time and labor intensive than the light dark bottle method. So we want to use this new approach on both natural assemblages and on cultured phytoplankton species. So we thus needed a place to test the oxytherm on these natural assemblages. So if you look at this map up here, on the western side in the yellow box, we have the York River Estuary, which is a shallow, partially mixed system formed by the Pamunkey and Mattaponi rivers. And then on the east in this red box, we have Cherrystone Inlet, which is a tidal creek on the bay side of the Virginia Eastern Shore. And since the Brush Lab currently makes extensive measurements of productivity in both the York and in Cherrystone, we chose those locations and sought to contrast productivity between the two. So then we also work extensively on harmful algal bloom or HAB species in the lower York River. And since we often don't have enough culture to run the light dark bottle experiments, the oxytherm presents a promising alternative. So we also tested it on our two naturally occurring species, which are shown here and are commonly referred to as Marge and Alex. So like I said, these two harmful algal bloom species are natural in the bay, and they bloom on a near annual basis. So this image right here shows a bloom of Alex. And these species produce toxins that lead to fish kills and have the potential to harm human respiratory systems. So this work acts as a preliminary research for an ongoing study modeling the causes and effects of these species. So I had three overall objectives for the study. The first was to compare methodological approaches for measuring changes in dissolved oxygen. So this entailed comparing the light dark bottle method to the oxytherm. The next objective was comparing plankton community metabolism between an east and west tributary of the Chesapeake Bay. So this is comparing Cherrystone Inlet to the York River. And lastly, the third and final objective was to compare the photosynthetic rates of natural phytoplankton assemblages of the Chesapeake Bay to our two cultured species. So to go along with these objectives, I have a couple of hypotheses. First, I hypothesized that we would see similar results between our traditional light dark bottle method and the oxytherm method, since they both measure changes in dissolved oxygen in a controlled environment. Next, I hypothesized that there'd be higher rates of photosynthesis in the York River as compared to Cherrystone Inlet, since there's greater freshwater discharge in the York River and thus greater nutrient loading to fuel primary productivity. And lastly, I hypothesized that there'd be greater photosynthetic rates in the HAB cultures as opposed to natural assemblages, since HABs have the potential to increase primary production in an ecosystem, and cultures tend to have more concentrated or are more concentrated. So these maps show a zoomed in image of the field sites where we collected our water samples. So in the yellow box, we have York River, and on the red, we have Cherrystone Inlet. And we selected 3 stations from each of these sites. At every station, we collected surface water samples to bring back to the lab, and we used a YSI to measure in situ conditions. So onto the light dark bottle method, our typical approach to study production in estuarine systems. So when we got back to the lab, water samples from each of the stations were placed in light dark bottles seen here, where they were incubated in this light box seen here at in situ temperatures. So the light box, as you can see, has a light source on the bottom. So when we place the light dark bottles in the light box, all of the different bottles are receiving a different amount of light on a spectrum. And the spectrum of light resembles that of which we would expect to see in the field. So for each station, we had bottles in the dark and then at 10 different light levels. In the light, bottles incubated for 2 to 2.5 hours, while the dark bottles incubated overnight. So about 16 3/4 to 18.5 hours. So now for our oxytherm method. So we used a similar range of light levels, starting at the dark and then incrementally increasing the PAR or light level each time. So the incubations for each light level lasted 5 minutes. And I switched out to a new 2 milliliter sample for each light level. And this is because although the machine is supposed to work by keeping the same 2 milliliters in, it did not work on our first couple of trials. And so we thought maybe that the raw water samples from the bay were not concentrated enough. So we switched them out. So also to test the sensitivity of the oxytherm, we used both the raw water samples along with concentrated samples from both the York and Cherrystone. We concentrated them because we were concerned that there was not enough phytoplankton in the raw water samples. And we concentrated the samples through a filter and resuspend technique. And then with the leftover water from each station, we filtered for chlorophyll A and ran fluorometry on this machine here, which is a Turner Design 10 AU fluorometer to quantify the chlorophyll A. For our harmful algal bloom species, we had 3 reps per each species. So 3 reps for Marge, 3 reps for Alex, and we used the same dark and light levels that we saw on the previous slide. So for each rep, we used the same 2 milliliters across all the different light levels, unlike what we did in New York and the cherry stone. And this is just because, like I said, the oxytherm method is supposed to work with the same sample. And since the cultures are high in density and presumably nutrient rich, got it. So we also then filtered for chlorophyll A and ran fluorometry on the HAP species. So this graph represents the results from one of our light dark bottle incubations. This is what we call a photosynthesis irritance curve, or PI curve, where on the x-axis par, or our measure of light, and on the y-axis we have photosynthesis, which all photosynthetic rates are normalized to chlorophyll to help us compare the different incubations, considering that there's different biomasses or concentrations in all the different incubations. So this curve is a nonlinear regression fit in the IBM SPSS Statistics 27 package using the Platt et al. Equation seen on the top of the slide. So let's explain this curve a little bit more. So this initial increase right here is it's almost linear. It's the light limited portion of a PI curve. So where with increasing light, we're getting more photosynthesis. Then we reach a point of maximum photosynthesis around here. And then we start to see photoinhibition, where after this certain point with increasing light levels, there's less photosynthesis. So in this graph, we have, once again, photosynthesis normalized for chlorophyll on the X or Y-axis and light on the X-axis. So this blue line is the same blue line we saw on the previous slide, which is the light dark bottle PI curve. And then on top of it, we have the orange line, which is the raw unconcentrated sample that we ran through the oxytherm. And the gray line is the concentrated sample that we ran through the oxytherm. So the general idea or trend that was shown in all of these, this is just for Cherry Stone Station 2, but it represents what we saw in all the other ones, is that there was no relationship between the PI curves for the light dark bottles and the oxytherm incubations. So I do want to note that in most of our graphs, the gray lines and dots were more, or the concentrated samples were closer to the light dark bottle values than the raw water samples, which is important for later. So this is the results from our Alex and Marge, running them through the oxytherm. And so once again, y-axis photosynthesis, x-axis is par, and the different colors represent the different trials for each species. So from this, we got well-constrained PI curves with the parameters that we talked about earlier, in most cases, with the exception of Marge run 3. And you will notice that most of our values are negative. So negative photosynthesis values mean that respiration is occurring. Thus, there are high rates of respiration in these samples that we ran through the oxytherm of our HAB species. So in this graph, this is moving back to our objective 2, so comparing the Cherry Stone or Cherry Stone stations to the York River stations. So we have the station numbers on the x-axis and photosynthesis, or mean photosynthesis, on the y-axis. And mean photosynthesis was calculated by using the parameters from the PI equation that we saw on the previous slide, and averaging rates of photosynthesis across a range of light levels. So, and I do want to note that we saw a similar trend when we graphed the max photosynthesis as well. So the takeaway from this graph is that we had greater photosynthesis in the Cherry Stone in the lower estuary, but there was greater photosynthesis in the York River in the upper estuary. And now moving on to our third objective, which was looking at the HAB species as compared to the natural assemblages. So the y-axis is the same with the overall. And then the x-axis from the bars represent Cherrystone, the York River, Marge, and Alex, with the error bars representing plus or minus the standard error, the mean. So the takeaway from here is that there is greater photosynthesis in Cherrystone and York River than in the HAB cultures, and that there is net respiration, see this negative bar right here, for Marge especially. So just to reiterate and recap what we just saw in these graphs, the light dark bottle and oxytherm produce different PI curves. The traditional PI curve shape was present in the HAB cultures that we ran through the oxytherm. And we also saw with the negative values that we got that the HAB species respired more. We also saw different photosynthesis in the lower and upper bays for the Cherrystone and York River stations. And then we saw more photosynthesis in the natural assemblages as compared to our two HAB species. So moving on to discussion. So back to our first objective, which was, will the oxytherm give us similar results as our traditional light dark bottle method? And so for natural assemblages, the short answer is no. However, for our cultures, the PI curves from Alex and Marge in this preliminary study looks promising. So that's exciting. So I used the overall slopes for each light level to calculate and determine changes in oxygen over time. But this is a machine, we're still learning it. So the first minute of our 5-minute incubations tended to have a higher rate, while the last 3 minutes tended to plateau more. So using, we would have used a different one. The PI curves could have looked different. So this is something to consider in the future. And also, like I said, our whole hope with this study was to see if this would be more time and labor or less time and labor intensive than the traditional like dark bottle method. But unfortunately, it was not. So as you can see, light dark bottle method usually takes 7 to 9 hours, but with those incubations, you can walk away and come back. Whereas the oxytherm method took me 8 to 12 hours, pulled a couple all-nighters, which after morning the field is not ideal. So I don't know if we can say this is less time and labor intensive. Also, you can see this is from my all-nighter. I was very excited to finally leave the lab at 5 a.m. So on to our second objective, comparing Cherrystone and York, our east and west tributaries. The upper York River had lower estuary and Cherrystone had greater photosynthesis than those in the York, whereas the York River upper estuary had greater photosynthesis than that of Cherrystone. And this could possibly be because, so that was station 3 in the upper York. There's greater freshwater discharge there because that's where the Pamunkey and Mattaponi rivers converge. So that could have impacted photosynthesis. For our third and final objective, so we saw greater rates of respiration in our harmful algal bloom species, especially in Marge, and then greater photosynthesis in our natural assemblages. So what can explain this respiration? Well, it could depend on what stage the culture population is in, whether the population is increasing or if they've hit their carrying capacity, which depends on when their media was last changed. Similarly, both depending on when their media was changed, and since we use the same 2 milliliters throughout the light levels, it's possible that these samples became nutrient limited, which would have limited primary production. So in the future, to improve this study, since the preliminary oxytherm results with the HAB species look promising, we can try using more concentrated samples of the natural assemblages to see how that does. Also, for the second objective, when we compared Cherrystone and the York River, we only had 1 day's worth of data to compare, so more days would have made this a stronger comparison. Similarly, the York River is massive as compared to Cherrystone, so we saw a much larger salinity gradient of 9 to 20 parts per million, while in Cherrystone we only saw 23.1 to 23.2. So choosing locations with more comparable salinities would also make this a stronger comparison. As I mentioned before, the growth stage and stages after medium could have media change in the HAB cultures could have impacted the results. So in the future, we can compare these cultures at different stages in their growth and examine the nutrient concentrations available. Since the light dark bottle method requires a large volume of water and cultured populations are limited, we have not been able to run the HAB species in the light dark bottle method like we did with our water samples from the York and Cherrystone. So this would be an interesting and important comparison to use against our oxytherm. And lastly, we could look at the different concentrations of HAB cultures, which is identified by cell counts. So I would like to thank my mentors, Dr. Mark Brush and Stephanie, for your support and guidance. Thanks to the Brush Lab, especially Sarah, for support in the lab and in the field and for providing me with Cherrystone data. Thanks to Emma for her help in and around the lab. Thanks to Doctors Kim Reese and Sylvan for providing me with cultures of Alex and Marge. Thanks to Hunter Walker for letting me use the nitrogen tanks. Sean Fate for driving the boat in Cherrystone. And thanks to our REU coordinators, TAs and directors. And thanks to the rest of my REU cohort for cheering me on and inspiring me every day. Any questions?
Smitty, I think we only have time for maybe like one question because we are running short on time, but feel free to ask Smitty any questions outside as well.
Hi, Smitty. Great talk.
Hi, friend.
So I know that the two methods, they have a time differential for how long they run, but do you think one provides better results over the other, or do you think there's a way that you could test that?
So from this results, the light dark bottle method provided better PI curves and PI curve parameters than the oxytherm did. So again, that could be because of how concentrated the samples were in the oxytherm, but yeah.
Righty, thank you, Smitty.
Right, well, that concludes the presentations for the morning half of our program. So I just wanted to give a big round of applause for all of our morning speakers. Great job. Good job. So now we're going to have a lunch break, and the students, we can meet over in the galley, which is around the corner behind the classrooms. But right before we do that, I wanted to get everyone together and we'll go take some photos out looking over the water. And then if everyone can come back here at 1:15, our next speaker will be at 1:20 promptly. So we'll see you back here at 1:15. All right, welcome back to our afternoon session of the Research Experiences for Undergraduates presentations. And we're going to have one of our interns be the moderator for this session. It'll be Eli Whitehead-Zimmers. And we're also, just a reminder for those who weren't here in the morning, we're going to have each student give a 15-minute presentation, and then there will be about 5 minutes left for questions. And if you're on Zoom, if you could please type in your questions to the chat, and Stephanie will read them out and we'll get them answered by our speaker. So we are ready to roll. I'll pass it on to Eli.
Thank you, everybody. Good afternoon. Thank you so much for coming out today for the second round of our review talks. Our first presenter of the afternoon is hailing in from the great state of New York. He's a marine biology major from the University of Miami, and he's here to talk to us a little bit about pearl production. Ladies and gentlemen, when he's not counting pearls, he's shredding on the guitar. Put your hands together for James Liu.
Thanks for that introduction, Eli. Hi, everyone. I'm James, like Eli mentioned. I come from the University of Miami in Florida. I was co-advised by Dr. Ryan Carnegie and Dr. Robert Isdell, and I'm going to be talking to you today about my study, which was called The Impact of Shell Length, Proximity to Water, and Density of Conspecifics on Pearl Production in a Stressed Quinquentia demissa Population. Okay. Ecosystems provide many ecosystem services to us humans. Ecosystem services are defined as benefits that we get from healthily functioning habitats. And so the Atlantic salt marsh, which is diagrammed here, is an example of an ecosystem that provides us benefits. A few examples of these ecosystem benefits are storage of blue carbon, acting as a nursery habitat for juvenile fish and invertebrates, and most importantly, protection from coastal erosion and storm surge. By some estimates, these wetlands provide almost $2 million per square kilometer a year in the US alone. Ecosystems are highly interdependent and interconnected systems, which means that when one link in the chain is impacted, the whole rest of the ecosystem can feel the results. And the link in the chain that I'm going to be focusing on for my presentation is the ribbed mussel seen down here in the low marsh. The ribbed mussel, also known as Quinquentia demissa, is an essential component of the salt marsh. It enhances cordgrass growth, which is one of the primary forms of vegetation, improves water quality by filter feeding, and overall contributes to a more sustainable and long-lasting habitat. And because it's such an important component of the ecosystem, it's important to understand and address any and all threats to the species that can be ameliorated. And an example of one such threat can be seen right here on the right, an excessive pearl production in a single population of the ribbed mussel found just 5 minutes away from VIMS. Normally in a stressed ribbed mussel, you'll see 50 pearls max. So when my advisor, Dr. Robert Isdell, discovered this back in 2014, he was very shocked. This has never been documented at this scale in the literature before, so we're dealing with something pretty unique. Pearl production in the mussel is an immune response, is often a sign of stress, and can even be a precursor to death. So when they first sampled this population, brought it back to the lab, they found that a living parasite is probably the cause of this pearl production. And another thing that they noticed in the initial observations is that there's a huge variation in the pearl response across the site, meaning that over here you might find mussels that have no pearls, and over there you might find mussels that have upwards of 2,000. And so this huge variation in the pearl response, both counts of pearls and size of the pearls that they found, is the subject of my study. Which brings me to my objectives. In the bigger picture, I aim to better understand a potential threat to ribbed mussels. Like I mentioned, they're very important for protecting the Atlantic salt marsh and are foundational to that habitat, but are also a potential conservation tool with their water quality improvement capabilities. There have been studies where they've put rafts covered in ribbed mussels in the water and found that they significantly improve water quality in the Hudson River. On the finer scale, for the purposes of my experiment, I'm aiming to understand and analyze environmental factors at the site, at the marsh, that could be causing this variation in pearl production across the site for both size and count. And I think that analyzing and understanding these environmental factors will give us more information about the pearl response and how to proceed conservation-wise, as well as give us a better understanding in general. And so I looked at 3 environmental factors at the experimental site. that I thought would probably correlate with pearl production. The first of which is the size of the mussel that I collected. I hypothesized that a larger mussel would probably contain more pearls and larger pearls. And the thinking behind this is that a larger mussel is an older mussel, and so it will have had more time for the parasite to be exposed to it, and will also have had more time for the pearls to grow and bioaccrete. They're also just bigger targets. The second factor that I was looking at is distance of the mussel from the water's edge in the marsh. I hypothesized that distance from the water would positively correlate with pearl counts, because as you get further away from the water's edge in a marsh, you also go up in elevation, and that increase in elevation translates to more abiotic stressors, such as thermal stress, desiccation, less feeding and settlement opportunities, and more. Just a generally harsher environment. Finally, I thought that mussels that were surrounded by more of its kind would also have more pearls due to increased intraspecific competition, as well as the idea that one mussel in a clump gets exposed to water containing a parasite, so will the rest of those mussels. And before I jump into my methodology, I want to stress that in modeling our statistics and designing our experiment, we had to keep a few things in mind about our variables. The first thing is that density of conspecifics and distance from the water are inherently linked and not independent of each other, because as distance from the water increases, you're going to find less mussels in the high marsh due to the increased abiotic stress. So that was something we had to keep in mind and factor into our models. And the other thing is that we have two latent variables, the first of which is quadrat placement, which was part of my sampling methodology. We believe that where you place the quadrat in the marsh definitely matters. Like I mentioned, there's differences in the mussels pearl production based on where you are in the site. So we factored that in as a latent variable, which means we didn't have numerical data to corroborate it, but instead treated it as something we knew had statistical or observable, I should say, impact. And finally, we thought that there could be a latent variable impacting both distance from the water and density, just something we couldn't think of that we wanted to keep in mind. And I believe that the combination of these factors would come together to produce an appreciable impact on pearl production. So this is the study site. This is a food line, and this is a movie theater. You can see that the parking lot kind of drains into the marsh that we're looking at. So that's something to keep in mind for water quality purposes. And then here's a even closer view. This is right in our backyard. This is a Google Maps view. We're right here at VIMS, just a 4-minute drive up US 17. So it's very local. Here you can see where we placed our 4 transects, which were part of our sampling methodology. They were 10 meters in length, evenly spaced about 5 meters apart, going from the water's edge up the marsh. Here's an even closer look. This is the water's edge down here. And this is the cordgrass I was talking about. And then here's me carrying out the sampling methodology. You can see the tape measure here, which was our transect, 10 meters in length. And along each of these 4 transects, we placed 5 evenly spaced 1/4 meter squared quadrats. In each quadrat, I recorded the GPS coordinates, counted all the mussels, and collected 2 mussels, one small between 25 and 50 millimeters, and one large greater than 50 millimeters, which came out to 40 mussels. Here's an example of high density of conspecifics. You can see tons of mussels are just aggregating. And so you can imagine if this mussel gets hit by water containing a parasite or is experiencing some abiotic stress, the rest of these will also be feeling that. So once I collected those 40 mussels, we took them back, weighed them, shucked them, which means get rid of the shell, and recorded the length, width, and height of the shells. After that, we took the tissue, dried it out in this little oven here, weighed that, and then dissolved it on this hot plate in beakers containing 10% KOH solution at 90 degrees Celsius. And from there, the idea was we had the mussels in solution, but the pearls would stay behind, and then we could filter them out using this apparatus, high-tech beaker and filter. And then finally, we would have all the pearls collected, and this is what they look like, just like little grains of sand. From there, I had to count and size them, and there were 13,609 of them, so it took a long time. And the only way I was able to accomplish it was using this setup you can see above. This is a Sedgwick rafter cell, which is basically a regular microscope slide with a lifted compartment that allowed you to place particles in it without them falling out. And then I had a camera mounted over them to take pictures for imaging software. I used ImageJ. This is an example of one of the pictures I took. We put no more than 200 pearls in a slide at a time because any more of that, all the pearls would be touching and the imaging software would not be able to distinguish them as individual pearls. So another thing that I had to do was go in with a toothpick and manually separate all the ones that were touching as best I could, which took so long. The visual scale here is 2 centimeters, and which I set in image J. This is the edited photo. I cropped out the slide, made it a black and white image, and then from there, image J counted and sized all the particles. With all of that data on pearl count and size, I mapped those variables as well as density of mussels for each quadrat across the surface of the study site, and I'll show you that in a second. And I also generated ridge plots showing pearl sizes, distribution per mussel in correlation with distance from the water and size of the mussel containing the pearls. Finally, we created negative binomial models showing impact of the 3 variables I was looking at on pearl counts to show effect size. And before I get into the results, I just want to mention the genetics aspect of my work. We extracted DNA from the mantle tissue of afflicted mussels, amplified it using a general PCR assay, and unfortunately, with the goal of identifying what parasite it was, it didn't work. You can see that these are pretty weak and unclear results in the PCR, just because the general assay didn't encompass whatever parasite we were looking for. But this is an ongoing and inconclusive aspect of the experiment, and we will find out what the parasite is at some point. But on the bright side, we were able to conduct histopathological analyses of the tissue, which basically is just looking at the tissues under a microscope. These are cross sections of pearls containing the remnants of something that was living. And this analysis also generated this photo. This is a pearl no bigger than the size of a grain of sand. And you can see the remnants of something that was once living, and I believe it was a trematode worm, probably Proctoeces maculatus. That's our best guess because it's a trematode worm known for causing stress in mussels and is common. But we are waiting for a positive genetic ID on that. So these are my results. These are the mussel counts compared to just mapped across the surface of the site. And you can see the bigger circles, which represent higher numbers of mussels, are generally found closer to the water, except up here in the high marsh, but that's because there's a channel here of water. So that makes sense given the decreased abiotic stress. And this is a map of pearl count across the surface of the marsh. And you can see a pretty positive trend of more pearls with increasing distance from the water represented by the bigger circles. The trend kind of breaks down for pearl diameter though, which is not what I was expecting. I thought that there would be, on average, larger pearls closer to the water, but you can see at least in this transect, that trend is definitely not the case with smaller pearls further from the water. This is further corroborated by the ridge plots that we generated. On the x-axis, you have pearl diameter in millimeters, and then each of these little tick marks represents a single pearl. And on the y-axis, you have increasing distance from the water's edge. So this mussel down here was the closest to the water, and this was the furthest. And then you have the same thing for size. You have the smallest mussel down here and the largest mussel. What I had expected to see was an increase in the mean, sort of like this trend, if you follow my cursor. But you can see that pearl diameter really isn't changing with an increase in these variables. To even further visualize that, this is a ridge plot showing the effect size, which is just a quantitative measure of strength correlation, basically, of my parameters on the pearl diameter. And you can see that the mean, which is the dark blue line in the middle, is just about 0 for all 3 of these variables. It's a different story though for pearl count. You can see that density has pretty much an effect size of 0 and size isn't much better, but you do see an observable effect of distance from the water on pearl count. And so just to summarize what I just said, there do seem to be more mussels closer to the water, which makes sense given the decreased abiotic stress. It's not really a clear trend for pearl size, which I had not expected, but there is a pretty convincing trend of increasing pearl count away from the water. Like I said, not really much of a trend for pearl size distribution compared to distance from the water's edge or size. But what we do take away from this is that distance does seem to be impacting the pearl production. And what could this tell us? This could tell us that stress is strongly correlated with distance from the water's edge, of course, and that size and density are of much less concern. And I think that this is probably because the stress, the additional environmental stress, is compounding with the effect of the parasite on the mussel and is enhancing the pearl response just to add much more stress to the lives of these mussels. Like I said, it's pretty clear that pearl production increases under stress, and this, if anything, tells us that density or distance is the most stressful of these 3 factors. It's also interesting to note that distance impacts pearl count, but doesn't have an impact on the pearl sizes that you're seeing. And I thought that this could possibly be because the mussels are producing so many pearls that they're not really concerned about size of the mussel or size of the pearl, wrapping it up really tightly. They just kind of want to encase it in a pearl and get rid of it, kind of a quality or a quantity over quality issue. And finally, I'd like to say that it might be the case that water is helping with the parasite-induced stress because distance from the water seems to be the biggest thing impacting it. And it might not just be because of the abiotic stress, but maybe the water itself is playing some sort of role here. So yes, I would like to thank all of these people for their help, especially my advisors, Robert Isdell and Ryan Carnegie, for making me feel like a part of both of their teams and including me. Dr. Grace Massey and Rochelle Seitz and Stephanie Peart for organizing and overseeing the program and making all of this possible. And finally, the National Science Foundation for the funding. And so that's it. And if anyone has any questions.
Wow. Thank you so much, James, for that incredible talk. We'd love to take a few questions at this time.
Hi, great job.
Thanks.
I was just curious, of the 40 mussels that you took, did any of them not have pearl production?
Yeah, actually, that was the most common thing that we saw. There were basically 3 categories of mussels that we collected. Mussels that had no pearls, and there were like 23 of them, like 23 or 25. Mussels that had a few pearls, like less than 70, and there were probably about 4 of those. And then the rest of them had tons of pearls, between like 200 and 2,000.
So was there like a threshold of like where you saw like pearl production? Does that make sense? So like, was there like a max value and then it like went up?
Those were pretty clear bins that we found, just, none, a few, and then a ton.
Okay, and a ton is like hundreds, thousands?
Yeah, the most I found in one was 2,361. And that doesn't even count the ones, the pearls that were embedded in the shell that you can't count. So it's even more, yeah.
I'm just curious to know how your lab people found out about these mussels behind the foot lion.
It was part of my advisor, Robert Isdell's thesis work. He was sampling sites, just looking at mussels, and he stumbled upon this one, opened it up, and found all the pearls.
But what made them choose the foot lion?
It was just a site, I guess, because it was nearby. Probably a better question for Robert.
Fantastic talk.
Thanks.
I had a question on the one of the latent variables in your earlier slides.
Sure.
It looked like where you put down the plot was a random effect, and I just wanted to clarify that. Was that the 0.5 meter by 0.5 meter plot? That's a random effect in your model?
Yes.
Awesome.
Yes,
okay.
it is.
Cool.
Any other questions at this time? All right. Oh,
You did such an amazing job. Good
Thanks,
job,
Lisa.
James. I have kind of a random question. I know I asked you for the perspectives, what the smallest one that you had found, but what was like the biggest pearl you had found? Like, was it?
Let's go look.
That big?
Almost 1.5 millimeters.
well, with that, ladies and gentlemen, let's give another round of applause for James Liu. Thanks for that talk. Our next presenter from the state of North Carolina is a bio and stats major from the University of North Carolina, Chapel Hill. And he's here to talk to us today about the Eastern oyster. Ladies and gentlemen, he knows his stats. It's about to get technical. Put your hands together for Garrison Bullard.
Hi. So
as Eli said, I'm from UNC Chapel Hill, and I was co-mentored by Gabby Saluda and Dr. Ram Lipsius, and I'll be talking about Eastern oyster body condition across varying habitats. So, as many of you know, Eastern oysters are an incredibly important resource for people in and around the Chesapeake Bay area and up and down the East Coast. In 2020, Virginia alone produced over $37 million of oysters in their commercial fishery. And oysters also provide an important habitat for many species, including commercially important ones such as striped bass and feeding grounds for blue crabs and other iconic species. On the right here, you see a shell that we pulled out empty from one of our study sites, and it actually had a blenny, which is a type of fish, living inside of it, and the texture that you see on the inside of the shell there are all of the blenny's eggs. So clearly it's a very important habitat. But unfortunately, in the past century or so, we've stark decline in Eastern oyster populations in the bay. This is due to overfishing that started in the late 1800s, as well as a couple of diseases that became prevalent in the mid 20th century. And current populations are estimated to be 1% approximately of what they used to be. So, of course, restoration and conservation of oysters in the wild is a great point of emphasis for the state of Virginia and other places up and down the East Coast. And to assess whether an oyster reef is restored, we have a general set of guidelines that are set for this location, the Chesapeake Bay, and they're based on expert knowledge, and they include threshold and target densities in addition to a couple of other metrics to assess an oyster reef's condition. So the threshold is considered 15 grams of dry oyster mass per square meter of reef and 15 individuals per square meter of reef. The goal is 50 of each of those metrics. So once you've hit 50, the reef is considered restored. But these metrics are nice round numbers, and they are based on expert knowledge, but they're not based on specific scientific studies. And they lump all oyster reefs in the bay as one, under one set of metrics. But in reality, the literature tells us that there's great variation between oyster growth patterns between different parts of their range and between different habitats. For example, some oysters grow in the subtidal zone where they're exposed to air for part of the day, and then some oysters are intertidal, or sorry, reverse. Some intertidal, they're exposed to air for part of the day, and subtidal, they are able to be in the water and feed all day. And there are also different types of habitats, such as natural reefs, like the one on the left here, which you can see us sampling, and the one on the right, which is on some bulkhead. It's artificial, and these two are treated under the same restoration metrics, but should they be? Well, I wanted to find out. So you might wonder, the metrics are based on densities. What does density have to do with the restoration of an oyster reef? Well, higher oyster density means higher larval recruitment, because oyster larvae use chemosensory techniques or habits to find adults, because the adults, you know, they've been successful. They've found a place to live and they're surviving there. So what better place for a larva to go? The more dense your oysters are, the adults, the more larvae you're going to get there and the faster your reef is going to grow. And also, just higher density means more larvae are produced, and some of them are just going to stay there on the reef, settle, and become adults. But what about the individual oysters? How does their health affect the overall reef health? Well, oysters that have higher, quote, condition are known to have more fecundity, meaning they produce more larvae. So what is oyster condition? Well, oyster condition is simply a proxy for oyster health, and there are a couple of different ways to calculate it, but in general, it is a metric comparing the overall biomass of the oyster's body to its size, whether it's shell volume, shell height, or something like that. So I used dry weight over shell height, which gives you an idea of, okay, this oyster has put so much energy into growing its shell, the shell height, but how much energy has it put into growing its body? So higher condition index means higher oyster health in a general sense. There are nuances to that, but I wanted to know, how does this condition vary by the oyster density, which I measured as biomass per square meter rather than individuals. And I'll be using those two terms interchangeably. And how does it vary by tidal zone as well? And is the condition versus density trend the same in the intertidal region as it is in the subtidal region, or is there a difference? So to find out, of course, we collected samples. Where did we collect samples? Well, we collected 3 samples in high density zones, which were loosely considered more than 50% oyster shell coverage, and 3 in low density zones. And we did that for each the intertidal and shallow subtidal zones, meaning 12 samples per site. We had 4 sites, 2 in each Timberneck and Cedar Bush creeks, which are both found in the lower York River near VIMS. Well, what was a sample? A sample consisted of haphazardly placing a quadrat somewhere on the oyster reef and collecting all of the oysters found within the quarter meter by quarter meter quadrat. An example is shown in the top left here. You can imagine those shapes are oysters. And on the right, you can see one of our collaborators from the archaeology department at William & Mary collecting a sample. Here's a faraway view of our sample sites. And here's a close-up view of our Timberneck sites. Each of these points is one sample, the red ones being subtidal, blue being intertidal, 1 through 3 being low density, 4 through 6 being high density. The exception being Timberneck 1, everything was high density. There were lots of oysters there. And here is our Cedar Bush sites. And so once we collected these samples, we brought them back to the lab for processing. We measured the length of all of the oysters, which is defined as the distance from the umbo or the hinge to the farthest point away on the oyster shell, because oysters don't exactly grow in a straight line all the time. And we measured the wet and dry masses of select oysters from each sample, making sure to encompass the entire range of sizes in that sample. And that's of the actual body of the oyster, not the shell. So since we were limited by time and resources, of course, we didn't biomass all of the oysters, but to make up for that, I constructed a log biomass versus log shell height plot seen in the top here for every single sample. And then I predicted the biomass, the dry biomass of the oysters that I didn't directly weigh based on their shell height. Then I used all of these biomasses, the predicted and the measured ones, to calculate the biomass per square meter in each sample. So using all of this data, I performed a generalized linear model comparison, predicting the condition index by biomass per square meter, tidal zone, and the creek that they're in. Here are my models. They were all fixed effects GLMs, generalized linear model, under the gamma family, and have a good suite of combinations of our predictors for which to compare. So our results are found here. The weight column is a proportion. It's saying that, okay, assume one of our models here is the correct model. It's exactly what happens in nature. What is the chance that it's this particular model? So the null model has 0. So there's essentially no chance that the null model is the true model. Now GLM3 has 0.55. So it's saying that there's a 55% chance that GLM3 is the most likely to be found in nature. Well, let's figure out how much better it is. So I compared the models based on a chi-squared test, and essentially what we're doing here is the larger model. If the larger model on the left is better than the smaller model on the right, then I'll have a very low p-value. If it's not significantly better, then I'll have a high p-value. So I compared GLM3, our highest weighted model, to the null model, meaning nothing matters. It's just constant between all of our variables, and the global model, meaning everything matters. And all of the interactions between the variables matter too. So that's our most complex model. And then I also compared it to GLM5, which was our second highest weighted model. And as you can see, the p-value for the comparison to the null model is very small, meaning GLM3 was better. The other two, p-values high, meaning the global and GLM5 models were not better than GLM3. So we're settling on GLM3 as our winner. And let's look exactly at what GLM3 tells us. Well, our intercept, which would be the average for Cedar Bush Creek, is 57 grams per millimeter. Timberneck Creek has an average of 18 grams per millimeter condition higher. And you can see on the right a plot of essentially our residuals versus the creek. And if you took a representative oyster from Cedar Bush, 57 grams per millimeter condition, well, that you would expect a similar amount of 38 gram per millimeter oysters from Timberneck. That's a 27% difference between creeks. This is a similar comparison for biomass per square meter. Well, our coefficient found in the table is negative 0.03. That's a little bit abstract, so I'll put it in perspective. This is saying that as the oyster density increases, the average condition of the oysters in that plot is going to decrease. And if you took an oyster, for instance, with 57 grams per millimeter in the lowest density, well, that's comparable to, say, an oyster with 39 grams per millimeter condition in the highest density, which was about 600 grams per meter squared, greater than the smallest one. That's a 37.5% difference. So very great difference between the two. We see an even stronger difference between the intertidal and subtidal zones. The intertidal zone is better, has better condition on average than the subtidal zone, seen by the negative coefficient there in the table. With that same comparison, we'd see a 40% difference in condition between the intertidal and subtidal zones. However, GLM3, if you don't see any sort of interaction term there, so this is saying that we don't have enough evidence to support that the density trend varies between the intertidal and subtidal zones. So Intertidal oysters have better condition. Why is that? Well, there are a few options. One is maybe the intertidal zone has better substrate, and that leads to better condition. After all, a lot of the subtidal oysters we were collecting are sinking in mud. They're covered in black anoxic mud for the bottom third or so of them. If a third of you is in mud, you're probably not doing too hot. You can't feed with your entire surface area, and there might be other stressors associated with drowning in the mud. And subtidal oysters might also tend to grow up faster to avoid burial in this mud, or other differences in shell shape might be happening due to stressors in either environment. Also, it's worth noting that there's higher predation and parasitism in the subtidal zone because they're exposed to the water all the time. So their main predators, such as blue crabs or parasites such as pea crabs, can access them much more easily. And the intertidal oysters also really have a need to retain moisture and food because, think about it, they're exposed to the air for hours per day potentially, where they can't feed, they can't get water, they have to seal all of that in. And so they might be better, they might need more body mass or need more fluid inside of them that the subtidal oysters simply don't have to worry about, and that could affect our condition. Now, we see that low densities have greater condition than high densities. And this is supporting the hypothesis that competition outweighs facilitation for these oyster populations. Facilitation meaning the positive impacts that oysters have on each other. So one aspect that has been cited in mussels is that of thermal buffering. A big clump of mussels together is a lot more insulating, almost like penguins huddling, than a single oyster is by itself, or a mussel is by itself. And this could be applied to oysters. And the fact that we didn't see an interaction means that, as far as we know, the competition and facilitations are applied equally across both tidal zones. So oyster habitats are different, and we've gotten more into the details of how they're different, but it's not for me to say whether this evidence is enough to change any sort of policy, but it is definitely food for thought for those making the policy. And potentially we could have further investigations on the exact densities needed for these conservation metrics, and whether those densities should be split up, whether the differences are enough for them to be split up, or whether the differences I found here are negligible in the grand scheme of things when compared to other sites as well. And it's worth noting that we've actually gone out and sampled 2 more creeks, 4 more sites, and collected just as many oysters, biomassed all of them this time instead of selectively biomassing. And we're still waiting on the results, the data to come in from that, but hopefully that will support our conclusions and lead to stronger emphasis on our results. Another thing worth pointing out is that random sampling is difficult to come by, both in the biomassing and in collection, but our new sampling should be able to confirm that, or hopefully be able to confirm that our initial results were not skewed by that bias. So finally, the obvious next step is to apply this research to different reef types, such as artificial bulkhead, riprap, and more. But I'd like to acknowledge my mentors and my lab here at VIMS for helping me with field and lab work, as well as my collaborators with the William and Mary Archaeology Department and my funders at the NSF, and everybody here at VIMS for hosting the REU program. I'll open up to questions now.
Unfortunately, thank you so much for a great talk, Garrison. We're running a little bit behind. We don't have time for any in-person questions, but if you had any questions or thoughts you'd like to share with Garrison, you're more than welcome to contact him via email. Thanks again. Another round of applause for Garrison. He did great, guys. Thank you. Up next, coming to us from the beautiful city of Philadelphia, is ecology, evolution, and biodiversity major from Temple University. She's here to talk to us about oyster substrates today. She really knows her stuff. She's worked hard this summer. Get ready for an insightful talk from Sarah Vadisher. Put your hands together for Sarah, guys. Thank you.
Is this thing on? Yes. Okay, great. Hi, everyone. My name is Sarah. I am working with Dr. Rochelle Seitz, Katie Nick, and Janina Patel, and we are going to look at artificial oyster substrates versus bare sediment. So we all know that oysters are very important, as we've seen from the past 4 presentations, and as Garrison mentioned, there's a lot of improvement for working with artificial reef types. So what I would like to do is look at biodiversity of oyster reefs. And for our purposes, I'll define biodiversity as the combination of species richness, or the amount of species, and species evenness, or the amount of individuals per species in a community. Increased biodiversity promotes ecosystem functions, which, as mentioned before, are ecological processes controlling energy and matter in a system. And then those ecosystem functions lead to ecosystem services, which are those benefits to humanity that we've mentioned previously. The Eastern oyster is a species that creates a 3-dimensional ecosystem harboring biodiversity with their reefs. And because of their productivity with nursery habitats, water filtration, and shoreline erosion protection, for instance, the Chesapeake Bay was once one of the most productive estuaries in the world, but now we're at essentially 1% of the native population. So a lot of those services are no longer present in the estuary. There are efforts to fund restoration and preservation projects of these native reefs, but ultimately what we need to look is to see whether artificial reefs and their different substrates can actually create the community that we need to rebuild. Certain material types like oyster shell has been proven to be successful for oyster recruitment, but we need to actually quantify the community structure. So my goals for this project are to measure biodiversity between different artificial reef types and bare soft sediment in the York River using biodiversity indices. Through this, we'll be able to explore how species interact with artificial reefs and work towards rebuilding the Eastern oyster population. Additionally, recognizing which reef substrate shows the significant differences in community recruitment and biodiversity is important for how we actually build these reefs. So in this graphic, you can see that there's 2 types of macrofauna that I'll be looking at. Epifauna are any sorts of animals and species that occupy the sediment surface, like mud crabs and oysters, while infauna are the more burrowing species like clams and worms. Each type interacts with oyster reefs. So let's see. For my methods, we had 4 different sites, and they were all in the York River near Gloucester Point, Virginia. So from going north to south, there's Cedar Bush at control 2, Indian Fields at control 1. And then as we get to VIMS campus, which is located by the star here, there we go. Impact 1 is VIMS Beach at Boat Basin, and Impact 2 is Andrews Beach right behind Andrews Hall. We sampled each site in 2021, just the bare sediment using suction sampling, and then we deployed a total of 36 reefs at sites. And then through this, we analyzed, we analyzed 9 reefs for this project. So then with that, we had a total of 6 different artificial reef types. And for my purposes, we'll be looking at these 3, which are loose oyster shell, loose granite stone, and concrete castle. These types have been studied and for oyster recruitment, but like I said before, we don't really know what types of species like to actually occupy them. And Oyster shell has been commonly used for oyster recruitment, but we wanted to test how they function in terms of granite and concrete because granite and concrete are significantly cheaper. We scraped the reefs after a year of succession at the impact sites and then processed for organisms, and which were then counted and identified. So then we also have these diversity metrics, which macrofaunal density per square meter of river bottom is not a diversity metric, but I wanted to include it in this section because we are using density for biomass in the larger project. But we have as the diversity metrics, the Shannon index, which is essentially diversity. I'll explain that later. Species richness, as I said before, and species evenness. These were calculated through primer E, and then the density was just standardized through R. So a big part of this project is using the before-after control impact design, or the BACI design. This is so that we can track change over time at the control and the impact sites, regardless of location or time. So what we're looking to see is an interaction between year and location, and not the effect of either or both of them. This is just an example of how heavy some of the reefs were. This is not one of the reefs that we actually are looking at today, but as you can see, it takes a village to get them out of the water, and you can see all of that biomass on top of just all the living things on it. So for our first question is, to what extent does reef presence impact biodiversity? And then this is just a closer look of that same X reef structure that we're not looking at today, but it's a great example of all the types of things living on it. So for macrofaunal density, we can see a clear interaction between before, which is 2021, and the after, which is 2022 on the x-axis, with the density being on the y-axis. We can see that clear interaction in that there's an increased density of macrofauna at the impact sites where we deployed the reefs. There's a 40% higher density, which is pretty good in my opinion. And certain macrofauna dominated the density. So now we'll look between groups. Worms were the dominated organism of our samples, with 161 times more higher density at the impact sites than the control sites. So this is a very strong indication of artificial reef succession for worms. And then here's a few of the types that we found: the clam worm, and then two tube-building worms. Crabs were also shown to have the same clear interaction between time and site. And as you can see, there were no crabs captured during the before for the bare sediment samples, but that makes sense because they prefer reefs for foraging and nursery habitats. So therefore, seeing them at the impact sites makes sense. And then for bivalves, we don't really have as clear of a picture of what happened here, but we can still see that increase in density overall at the impact site. The low sample size of my project might account for the trend in the control sample, but that'll likely be resolved with the larger project. So while sampling, we found many ribbed and hook mussels, which are these guys, nested in the reefs, while the bare sediment contained mostly burrowing clams like Maya arianara. And we only sampled the reefs at the impact sites, therefore we might have not found as many of these burrowing clams, and there might be a whole bunch more. For the main diversity metric, the Shannon index, there's an interaction between time and location for diversity. The increase is visually not as dramatic as an increase because it's an index, but we can still see that there is an increase in diversity, which is what we were looking for at the impact site. The control and impact sites will likely have the same diversity in 2021 when increasing the sample size in the main project, but we can still see our general trend. So to summarize the past couple slides of our BACI analyses, reef presence clearly increases macrofaunal density and diversity. Our second question is, to what extent does reef structure and material type differ in biodiversity? So this is the inside of the castle structure when we took them out from the impact site. So you can see that there's a large clustering of organisms in those corners. Yeah. So as we saw before with the Bakke analyses, the impact sites containing the reefs display a clear increase in density compared to the control sites with only bare sediment after a year. The loose granite stones show the highest macrofaunal density of organisms, while the oyster shell and concrete castle perform similarly down below. This is most likely because granite stones have a happy medium for interstitial space, which is the area in between hard substrates and flat surface. So as you can see, there's kind of like the granite stones are heavier than the shell, but there also is more movement in between them than the castle. Shell also has a large amount of interstitial space because of the irregularity of the shell shapes, and they're lightweight, so the shells can move around a lot more. And the castles are obviously way more heavy, and they have much more flat surface area than the granite. So when using an alpha value of 0.1 to account for low sample size, oyster shell and granite stones show a statistically significant difference in diversity. These basket reef types show a higher diversity for the same reason why granite had the highest macrofaunal density, the ability to capture mobile macrofauna. So the baskets of the shell and the granite, they're containing the stones and the shells. Therefore, when we take them out of the estuary, we can actually capture any like fish or crabs that might get stuck in there, rather than actually needing to like haul out a reef and like shake off most of the things. So the slight lead in diversity shell has over granite is also predicted by literature, since oysters recruits prefer shell as sediment substrate. But if I didn't say it before, we're looking at just the year of 2022 now, because in 2021, the before, there were no reefs deployed, therefore there's no reefs to compare. And we just had the bare sediment of the control sites to compare. When looking at species richness, Castle shows the most distinct species out of the substrates, but all the reefs are significantly different from the control. This further reiterates that reef types impact species richness and need to be further investigated to see the degree at which material impacts species composition. The control sites show highest species evenness, likely because infaunal bivalves tend to compete less and fill the same niche in the bare sediment compared to reefs. As mentioned before, each reef type has a different amount of interstitial space, leading to differences in how organisms can utilize the shelter and move around within the reef. Shell has the most interstitial space between each shell, like I said before, because they can move around and there's more irregularity. Therefore, macrofauna have to navigate the shells around when disturbed, and different from the granite and the castle, which are heavier and more solid in structure. So that might lead to more evenness and less species clustering together. So now we're going to look at the first part where this is a different type of plot, and we're just looking at the first part where we're looking at whether there's a difference between the before, after, and then the control impact sites. This is the Bakke analysis again, but on a different plot. So what we're looking at is a non-metric multidimensional scaling, or NMDS plot, and it's showing time and site type. So time is the colors, and then site type is indicated by I or C, which means impact or control. The NDMS plots are an ordination plot based on the Bray-Curtis similarity matrix, accounting for the species composition similarity matrix. So essentially, the entire matrix is ranked based on the most similar communities, and then it's plotted onto a 3D scale. So the different axes for this one would be year, control, impact, and species composition. So essentially, when you're seeing this clustering, this is showing communities with similar species compositions, like the impact sites right here. And then the global R value is based on the analysis of similarities metric test, where for our purposes, anything above 0.35 is considered good and different. Therefore, we can see how the 0.608 also corroborates that year and site type are different from each other with this clustering. Then similarly, if we take away 2021, you can really see that there's a strong difference between the impact sites and the control sites. And then the R value is 0.927, and the stress value is also quite low, meaning that this is a really good indication that we have a true interaction here between how different the two sites are and how reefs make a difference in diversity. And then I'll wrap up my results with a comparison of species composition between reef types in 2022. So color here distinguishes reef types, and we can clearly see a kind of clustering of communities with the same reef types. And the global R value for the entire NMDS plot is 0.835, which is pretty good. And even better, we can see that shell and granite are completely distinct from each other with this test. So this is also a large indication that we really need to look into material types for different reefs to see what types of species are actually inhabiting them, because with this study, we can clearly see that they are different. So overall, we saw that macrofauna prefer to settle around artificial reefs compared to bare sediment through the drastic overall increases in macrofaunal density and diversity from 2021 to 2022. From the images of each reef after collection, so this whiter shell here is that new oyster shell that we had to input when we took out the samples. But you can see that they all have a bunch of stuff settled on them. Native reefs are an inherent ecosystem, so it makes sense that we would see organisms that like the natural shelter, foraging grounds, and nursery habitats that these artificial reefs stimulate. When comparing reef types, we saw that there's a clear distinction between each site and species composition. And then that clear distinction between shell and granite are also really interesting, and future studies using sediment traps, flow meters, and different basket materials might help understand the differences between that granite and shell composition. And then additionally, from the species richness of the reefs, we saw that there might be some differences in how species are competing, but that is also another metric that needs to be studied, and understanding that that dynamic is super important for restoring healthy oyster reef communities. In summary, reef presence positively influences density and diversity to a high degree, and reef structure and substrate type affect macrofaunal density and diversity. From literature, we know spaces with more interstitial space has more diversity, with oyster shell being the preferred substrate for oyster recruitment. But we can see that shell isn't the catch-all for community structure. Shell is an expensive substrate, so visualizing how most of the density came from the granite baskets actually really impacts what we need to do in the future for future initiatives. And then the most important takeaway from our work is that this is a completely new metric for understanding oyster restoration in the Chesapeake Bay. With secondary productivity as the currency of restoration, metrics like this are the first step to studying productivity and longevity of artificial reefs. Artificial materials degrade, therefore we need materials that can create that healthy oyster recruitment, which also creates the healthy communities that can then indicate ecosystem health. And with that, I would like to thank the Indigenous people of the Chesapeake Bay for their water and their land, my mentors, Dr. Rochelle Seitz, Katie Nick, Jeannie Patel for their guidance, my lab for field support, Gabby for statistical support, the RU mentors and the RU cohort for their support, and just a great time here, and then VIMS and NSF for funding. Thank you, everyone.
All right, we would love to open the floor to any questions at this time.
So I was really surprised by the fact that oyster castles had on average 2 more species for species richness than the other 2 artificial reefs. And I wonder if you know which species that was.
Yeah, so what we're thinking for the castles is because the castles have a really flat surface on them. Let me pull up a picture. Yeah, so as you can see, this really flat surface of the castle is really good for tube builders, which are worms. So we're assuming that it was worm species that were creating more of diversity in the castles, because not like crabs, bivalves, it's harder for them to stick to those surfaces. So that's what we're thinking.
Great. We have time for one more question.
I'll be quick. You did an amazing job there, by the way. So you had mentioned different types of substrate. Could you go a bit more into those? Thank you.
Differentiate them? Is that what you said? Oh, yeah. So the different substrates are just the loose oyster shell. So the difference between the reefs is just what makes them. So the loose oyster shell, the granite stone, and the castle, the concrete castle, are what the substrates are. So it would be the material types are like the oyster shell itself, the granite, and then the concrete is what makes the difference. And we also have the structure that makes things a little different. Like, as you can see, the baskets themselves, they have looser material types. So like the shell can move around a lot more. The granite is more firm, but it still has more less interstitial space in the shell. And then the castle is more rigid and it can't really break apart as well or move. And there's even less interstitial space because of that.
Great. Thanks so much. Let's have another round of applause for Sarah, everybody. Next in our program, we have a biology major from the College of William and Mary. He's going to be talking to us today about tethering assumptions in marshes. Ladies and gentlemen, put your hands together for the king of crabs, the master of memes, Cole Miller.
Thank you for that beyond flattering introduction, Eli. So as mentioned, I'm Cole Miller, and I'm work with Dr. Lipschitz and Challen Hyman on a simulated mesocosm experiment using Callinectes sapidus to test a tethering assumption in marshes. So for all of those who may be unaware, Callinectes sapidus, also known as the Atlantic blue crab, is a swimming crab found up and down the eastern seaboard in various marine and estuarine habitats. Here in the Chesapeake Bay, it supports a large commercial fishery, bringing in millions of dollars annually in revenue, jobs, and equipment. There's also a dominant benthic predator for other various marine invertebrates, such as worms, bivalves, and other crustaceans. And on top of serving as a predator, it also serves as a critical prey species for a lot of fisheries organisms, most notably the red drum. However, the blue crab recently has been seeing record low population levels due to various pressures, including climate change, overfishing, and habitat loss. This makes it an ideal candidate for study of the conservation of East Coast ecosystems, along with the proliferation of multi-million-dollar industries. So to kind of focus in where we are, I'm going to bring you to the life cycle. So after the females migrate to high saline waters, in our case, the mouth of the Chesapeake Bay, they spawn where the larvae spend about a month off the continental shelf before migrating up into estuarine nursery habitats and recruiting into juveniles. And these juveniles in nursery habitat is where we're focusing our study. So some of you may be asking, what exactly is a nursery habitat? Well, simply put, a nursery habitat is a habitat that produces more individuals compared to other habitats. And this is important because they allow the crab ample access to food and nutrients to grow, and as well as serving as vital protection for predators. It has long been described that seagrass, more specifically the Zostra marina species, has been the preferred habitat. However, due to rising induced temperatures in the bay, we're seeing die-offs of these beds. However, I would also like to bring your attention to salt marsh, particularly that of the Spartina alterniflora species. It's been shown, recent research has shown that it is a high viable alternative to seagrass, having high levels of production of juveniles equivalent to that of seagrass beds. They also have the added benefit of being far more abundant. And this combination of abundance and more resilience in face of climate change makes it an ideal habitat worth of more study. So when it comes to nursery habitats, one of our main things we're concerned about is the survival, particularly of our little crabs. So when we measure survival, there's multiple ways we go about doing that. I'd like to draw your attention to the two methods on the left, caging and mark recapture, also known as tagging. While commonly used, they both suffer from issues stemming from unnatural conditions and unrealistic assumptions, respectively. Our focus is tethering. While not a perfect simulation of natural conditions, it has advantages over the other two and is the primary focus of our study. So what is tethering? Well, tethering in essence is fastening a prey item to a weighted line, usually to allow you to go back after leaving it in the field to find it again. And it's been great because as opposed to caging, it allows for a wider range of movement as the line has a lot of slack compared to the weight. It also allows for natural behaviors from prey, more natural behaviors from prey and predators by having more minimal gear and extended human absence. However, there are a few caveats when it comes to tethering. So when you're observing rates of survival in tethering, the rates are relative, not absolute. This is because tethered animals die more. This is because while it has a wider range of movement as opposed to caging, they don't have an entire free range of movement. So therefore, they have to rely on methods such as hiding to escape predation. And this creates a negative survival bias. And we can actually quantify this bias and to help us find more absolute rates of survival. I've outlined this with an equation where the mortality of a tethered experiment is equal to the tethering survival proportion, which I've just listed as bias, times the absolute rate of mortality. And this image here just shows that we expect tethered individuals to die more often. Now, let's get on to the concept of habitat-specific interaction effect. So when you're doing tethering experience, especially across nursery habitats, this bias is assumed to be the same across all of them. So for instance, that bias coefficient would be the same in unvegetated habitat compared to, let's say, seagrass. Now, this comparison has actually been shown before. However, this brings us to the main crux of our experiment. Does this assumption hold true in salt marshes, especially considering the viability as an alternative nursery habitat? So we're seeing if that bias coefficient in marsh is equal to the ones established in seagrass and unvegetated. So the objective is to quantify the presence or an absence of a habitat-specific interaction effect in a marsh. And those who are still a little hung up on the concept of interaction effect, we're essentially seeing if the effect on marsh habitat on survival matters if the prey is tethered. So now that we've established the primary objective, let's dive into the hypotheses of this experiment. Note that all hypotheses in this section are defined with a model, with survival, denoted by the word survival, being a function of, with the function being represented by a tilde, of numerous variables that I'll get into with each hypothesis. So our first two are our baseline hypotheses built upon previous research. Our first one is that tethering, denoted by T, will have a negative effect on survival. Secondly, shoot density, denoted by SD, will have a positive effect on survival. Now I kind of got those out of the way. Let's get into the actual crux hypotheses at the focus of our experiment. Hypothesis 3, tethering and shoot density will have two separate yet conjoined effects under the same umbrella experiment in survival based on their respective hypotheses. So we just took the model and combined tethering and shoot density with a simple addition. Hypothesis 4, the tethering habitat interaction, that interaction effect, denoted by T times SD, will have an effect on survival. And under this hypothesis, we're testing that the tethering, that bias in unvegetated is not the same to the bias in the marsh habitat. Then we built some hypotheses based on natural variation we observed in our experiment. Our first one is that dissolved oxygen will have an effect on survival in conjunction with the interaction effect. We also have a sub-hypothesis without the interaction effect. Predator size, denoted by PW, will all have an effect on survival in conjunction with the interaction effect and dissolved oxygen. Again, please note that there's a sub-hypothesis without the interaction effect. So let's get into the methods of this actual experiment. So for actual tethering, we applied a pea-sized dab of Gorilla Glue between the rear set of walking legs and our prey blue crab. We then glued 25 centimeters of monofilament fishing line to the glue on the predator. We then attached the other end to a lead weight. We reinforced the line and glue with a square piece of duct tape, colored tan to avoid any flashy colors that might attract the predator. We then left the tethered individuals to acclimate in a private tank for 24 hours. That way they can get accustomed to having the tether on them. So the actual mesocosm setup. And for our case, a mesocosm is just a laboratory experiment we're using to simulate a set of natural conditions. So this is the actual experiment with all the factors at play. We have 8 tanks divided into 2 groups of 4 based on their tethering treatment, tethered and untethered, and I'll sometimes refer to that as free swimming as well. They have an ascending, each tethering treatment has an ascending level of marsh density, starting with an unvegetated representing by 0 shoots per square meter, low density 178 shoots per square meter, medium, which is 267, and high density, which is 356. So the actual tanks themselves. The water was pumped in from the York River and drained in the middle by a standpipe to keep water suitable for the specimens. Here you can even catch a glimpse of some of the ascending densities with unvegetated and the closest to the camera, increasing in density as you move further away. Please note that wooden dowels were used instead of actual marsh grass to reduce the variation in issues caused by maintaining living plants. So the actual tanks themselves, dowels were fixed to a pegboard kept 5 centimeters beneath river source sand. And here you can get a better view of the tank components, including the standpipe in the middle and the water input up top. Predators were randomly assigned a tank and left to acclimate for 24 hours before the trial. We use blue crabs as predators, as adult blue crabs are the primary predators of their own species. Up to 30% of an adult blue crab's diet is fellow blue crab. We then added our prey randomly and let the trial run for 24 hours to give ample time for feeding. Survival was collected by draining each tank and looking for any living prey. I will also note before each trial, we also record dissolved oxygen, temperature and salinity, which were not included in our final suite of models, prey size, also not included in our final suite of models, predator size, and survival. Survival being a binary number, a binary measurement of 0 meaning death or 1 meaning survival. All data analysis was done using the R programming language, and all models were individually evaluated by logistic regression. We then applied a method of finding statistical support using AIC. This method compares two given regression, any amount of regression models, with the lowest scored model being considered the most statistically supported. Models within 2 points were considered to be comparable with fit. So here's the results of our AIC scoring. It's kind of a lot to look at, but I'll kind of whittle down what you need to focus on. So we have our hypotheses, our model, and the formulas I outlined in our hypotheses section. We have our AIC score in that 4th column. However, since AIC score is a relative metric to our data set, we had to adjust it based upon the models we observed. So that brings us to our delta AIC column, which is the change in AIC relative to our best fitting model, which in our case is model 6, or that sub-hypothesis of the DO model. We then use this change to calculate weighted probability. The probability of this model being the best fitting model, given our data, assuming this is the only set of models we considered. However, since we wanted to quantify the habitat-specific interaction effect, we decided to compare some of our best fitting models to see if we can quantify the presence or absence of one. In doing so, we compared our top 3 models, our best model, the model at the bottom, and then the model, our best model, then our 3rd best model was our best model with the addition of the interaction effect. So we compared our models by a log likelihood chi-square test. In layman's terms, essentially this test is to compare the goodness of fit between 2 models given our data set. So for our first test, we compared our best fitting model to our best fitting model with the interaction effect, which was our 3rd best model overall. The number on the right shows that these models were not different in explaining our data set. So between the 2 of them, we used this one as a representative model. And because these models were similar in terms of fit, by proxy, we can eliminate that a tethering habitat interaction effect is not present, as is not contingent modeling survival. We then compared our best 2 fitting models, our compared model to the best model being our best model, just with the addition of predator width. Again, the number on the right shows that we have no difference between our 2 models. So again, for simplicity's sake, we decided to go with our already best model as our representative one. So applied our logistic regression to this model. And logistic regression found that chute density, tethering, and dissolved oxygen were all important predictors in survival. But I want to draw your attention to the estimate column, as this allows us to numerically quantify the effect of each variable. So what this says is that we take SD, chute density. What that number means is for every chute we add to a given tank per se, a prey item is 2/100ths times more likely to survive. A tethered individual, T, is 1.14 times more likely to die. And for every unit increase in dissolved oxygen, a prey item is 1.47 times more likely to survive. So let's visualize this data. There's no new ground is being made here, but essentially this plots chute density on our x-axis and probability of survival on our y-axis. Notice that the difference between our free swimming and tethered, denoted by red and blue respectively, and how tethered is overall less levels of survival. However, across both treatments, as we increase chute density, probability of survival goes up. Note that the juxtaposed points are the average probabilities of survival given our 4 density treatments. Next is our dissolved oxygen. Here we observe the relationship between dissolved oxygen and survival. And for the ease of visual simplicity, this is across all treatments. Higher levels of DO have higher levels of survival. That's all this is saying. But I'll explain our reasoning behind this trend in a few slides. So in summary, there is no habitat-specific interaction effect between tethering and marsh habitat. Chute density and dissolved oxygen were the best predictors in promoting survival. Tethering had an overall decrease on survival, and predator size had no effect on survival. So what can we take away from this? Well, that tethering assumption holds true in marsh habitat. Yay! So that means when we're doing cross-habitat analysis in nursery habitats, we don't have to do anything special with marsh as it has no influence on that tethering bias. Juvenile conservation should be focused on high levels of structure and dissolved oxygen as it promotes survival. And that DO point can be elaborated on as larval specimens of blue crab have been shown to be very responsive to dissolved oxygen. And that could be have some spillover into our smaller juveniles. In marshes, the size of larger blue crab predators doesn't influence predation. And this can be done to research published this year that shows that blue crabs in marsh prefer alternative feeding methods as opposed to just regular foraging. However, this may not hold true for smaller predators, including juvenile on juvenile cannibalism. For acknowledgements, I would like to thank my grad student, Challen Hyman, for taking me under his wing for this REU project and over the last year as a whole. Dr. Ron Lipschitz for taking me on as his REU student. Dr. Rochelle Seitz, Dr. Grace Massey, and Stephanie Pert for organizing this REU program and has accepted me as one of them. The Marine Conservation Ecology Lab, and I was told to plug #CrabLab. All my fellow REU interns, and everyone who contributed ideas and supported me through this experience. I'm happy to take any questions if you have any.
Thank you so much for that amazing talk, Cole. We'd love to take a few questions at this time.
Did you name the crab predators?
Thank you for that question that I totally didn't plant you in the audience to say. But yes, we did name our predators specifically. So you can play the game of pick your favorite. Yeah, they're all had their own individual personalities, very near and dear to our heart. So our personal favorite in the experiment was Emperor Claudius. You know, hard shell taco is also a favorite. So.
Hello, you did a good job. Great job. Which one did you hate the most out of all these crabs? Because I know they're
Oh,
very
did I hate the
aggressive.
most? I will say, well, that's a good question. I would say Reginald because he went out of his way to try and pinch me a few times. So yeah, he was also our biggest. So. Evil Bob, he was evil. His name, evil's more of an adjective than it is a name. But you know, I mean, blue crabs, they have a lot of personality. When I say personality, I mean they're really mean. So he was just mean.
Evil Bob pinched me on a Saturday morning after I moved to the backup tank, and he needed a name, so I was like, "Well, you're."
Looks like we have another question over here.
Yeah, hi Cole. Good job. I have a quick question, which may be easy or not, but why do you think predator size didn't actually have an effect? Like, why didn't larger predators want maybe to eat more than, let's say, a smaller one?
Okay, so let me bring this up. So all of our predators were kept between approximately 102 and 170 millimeters. So the research this year showed that in marsh habitats, blue crabs tend to opt for more of an ambush feeding, where they'll wait in a deep pool of water and wait for prey to come by. However, we can't reliably say that was used. However, even in our densest treatments, there was a few times I saw a crab able to kind of like navigate between it. So they all had, even some of the larger crabs had the ability to navigate in between even our densest chute treatments. So that's probably why it had no effect, because none of them were like, it could, like, yes, it deterred predation, but never truly encumbered them if they were like really looking to eat.
Any other questions? Well, with that, let's give another round of applause for Cole and that great talk. Well, ladies and gentlemen, thank you again for coming out tonight. We have only 2 talks left today for the rest of our program. And up next, we have with us presenting someone all the way from Arizona coming to Virginia this summer. She is a biology and philosophy major from the College of William and Mary. She's here to talk to us today about worms. Sure to be interesting. Put your hands together for our resident Myers-Briggs ENFJ, Alexandra Pomeroy.
Is it working? Okay, cool. Thank you for that very enthusiastic introduction. Like Eli said, my name is Alex. I'm very excited to share with you what I've been up to this summer. I've been working in the Fisheries and Aquatic Health Sciences department, so we got a lot going on here. But before I start, I wanted to point out our star of the show, a mature female blue crab, as she is pictured up here. I realized, because before I came to VIMS, I didn't realize what a pregnant female looks like. And so that large orange mass on her abdomen is her egg mass, which I'll refer to as a brood from here on out, but just to orient yourself a little bit to their anatomy before we begin. So, as most of us know, the Chesapeake Bay blue crab is very commercially and ecologically important to Virginia. However, over the past 30-ish years, blue crab landings have declined significantly. This could be the result of a number of factors: introduction of the invasive blue catfish, nursery habitat loss, poor recruitment, or just general disease in general. Among these factors, though, female reproductive output has not yet been investigated as a metric to assessing the changes in the population. Previous commercial crustacean fisheries have seen failures caused by high brood mortality, and much of that can be attributed to a nemertean worm. Nemertean worms, more commonly known as ribbon worms, house the genus Carcinemertes, which is a worm that predates on crustacean eggs and are commonly found in crabs. The species Carcinemertes carcinophila is found in our Chesapeake Bay blue crab. There you go. These worms feed and grow to maturity, mate, and lay their eggs all within the brood of their host female. When the female does have eggs, that's where the worms reside, but when she doesn't, they can migrate into her gills and encyst there, as that kind of a really concentrated picture of them at the right. All of those pink coloration that you see in her gills are nemertean worms. Since their identification in the 1840s, very little peer-reviewed literature has been published about the relationship between the blue crab and the nemertean worm. So that's why the objective of my project this summer was to quantify the effect this worm is having on blue crab egg mortality. So essentially, we wanted to figure out, one, how much are these worms eating? And two, how many are out there in our female blue crab population? In a hope, we were going to use these metrics to build and piece together the puzzles and build a better picture of what the relationship between these two organisms look like. In terms of hypotheses, I anticipated that the worms would consume a measurable percentage of the female's brood, affecting their reproductive output, and that worms of larger sizes would eat more eggs than those that are smaller in size. This has been seen, this relationship has been seen in other species of Carcinemertes, and so I expected it to hold true in this one as well. To briefly address the details of what our project looks like, to estimate the natural worm intensity in wild-caught egg-bearing females, crabs were collected by the VIMS trawl survey from the main stem of the bay, and the specimens to run our worm consumption rate trials were collected from VIMS trawl survey as well, but from the James River. All these individuals, once collected and caught, were brought back to the lab for processing. Once in the lab, the worms residing in the females from the main stem of the bay had their worms enumerated from both the brood and gills, and then those values were recorded for later analysis. The crabs collected from the James were weighed and measured, and then their eggs and nemertean worms were extracted from their brood and were placed in wells for acclimation. Once acclimated, both of those eggs and worms were placed in their new treatment wells, and photos were taken, and they were ready to go. Over the next 6 days, for every 24 hours, we checked feeding in all 60 wells. 48 of those were treatment wells, which contained worms, and 12 of those were controls that only had blue crab eggs. We checked for feeding by the presence of an empty egg case. An empty egg case was a reliable way to check for feeding because the worms use their proboscis to puncture the egg membrane and then suck out the yolk. So it was very easy to tell the difference between an eaten egg case versus an embryo that may just be developing poorly. Lastly, we also used the photos that we took during the processing stage to measure the nemertean worms using Fiji, which is a photo processing program. We also used R to do all of our statistical analysis. We used a simple linear regression to test the effect of worm size on consumption rate. We did transform the data just using the square root function to normalize it before doing this though. The other metrics that we use, though, we're going to test the presence of the worms in the bay. We used prevalence, abundance, and intensity, which are those metrics used commonly in parasitology. Prevalence being the total number of infected individuals over the total number of individuals from our sampled population. Abundance being the total number of worms over the total number of individuals from our sampled population, and intensity being the total number of worms over the total number of infected crabs from our sampled population. So that's a lot of stuff, but let's get right into the results, starting with objective 1. The effect of worms on egg mortality. So here we have a simple histogram of the total eggs consumed over the 6-day trial period. On our x-axis, we have total eggs consumed, and on our y, we have frequency, which is the number of times that value of eggs was consumed by a worm over that 6 days. So we have our total of 48 worms, which were our treatment wells, and on average, they ate about 3 eggs over the course of the 6-day period, with a standard deviation of about 5 eggs. However, not all of our worms fed during those 6 days. 73% of them were feeding. And so this is just that picture, that figure given again with just those worms that fed over the 6-day trial period. So we have a slightly larger average of about 4.4 eggs eaten over the 6-day period and a slightly larger standard deviation of about 5.5 eggs. We also looked at their consumption in terms of average consumption rate. So how many average eggs were eaten per a day over that trial period. We are looking again at all 48 worms, and on average, they ate about half an egg per day with a standard deviation of about 1/8 of an egg per day. And then again, we removed those worms that did not feed, and our new mean was about 3/4 of an egg per day and a standard deviation of almost 1 egg a day. Now we can look at the relationship between worm size and consumption rate. On our x-axis, we have our worm size in millimeters and our consumption rate on our y. And as you can see from our very small r² value and very large p-value, there is no significant relationship between these metrics. Now to address objective 2, we have a simple bar plot of prevalence of worms in the brood of individuals that we collected. On average, from the month of May to July thus far this summer, we had about a 40 or 39% prevalence. In June alone, we had a slightly higher prevalence of about 52% of individuals having worms in their brood. And in July, we had a mean prevalence of about 35%. In terms of brood abundance, we're now looking at a box plots just so you can get a visual representation of the dispersion of our data. In June, we had about 13 or 17.8 worms per a brood on average. And in July, we had about 16.1 worms per a brood on average, but you can see the dispersion there. We have almost 204 worms found in one brood, and that was about the maximum we've seen this summer thus far. But on average from May to July in 2022, there was about 15.6 worms per a blue crab brood. Lastly, to look at brood intensity, we saw about 28.6 worms per the brood of an infected individual thus far this summer. As you can see, May is no longer on the screen, but that's because we didn't collect any infected individuals in May. They just weren't there. And so in June, we saw about 27.9 worms for the brood of an infected individual. And then in July, we had 29.1 worms for the brood of an infected individual. So I just threw a lot of numbers at you, a lot of different ways to look at what's going on in terms of worm presence and how much impact they are having on egg consumption. So I'm going to try and put that into a little bit of context now. Let's look at some hypothetical case examples. So let's start with a conservative case and say that we have a worm that's only eating 1 egg every 6 days. And that brood that this worm is living in is developing over a 12-day period, which is on the shorter end of average blue crab brood development. Over those 12 days, that worm is going to consume approximately 2 eggs, which is really insignificant, right? Those broods can be up to 3 million eggs or anywhere on the low end of 750,000. So 2 eggs from that, who cares, right? But let's say that we're going to take the average abundance in the brood, which we saw was about 15 worms, and assume that all of them are feeding at this minimum rate of 2 eggs over the 12-day development period. Still, they're only going to consume about 30 eggs from that brood, and that's about 0.001% of the brood, which in the grand scheme of things is a drop in the bucket. So that's good news. No one has to lose any sleep tonight. But let's look at an extreme case. So on the longer end of brood development is about 17 days. So let's assume we have one of those really hungry worms and it's eating 27 eggs every 6 days, and the brood is developing over 17 days. Over that period, this single worm could consume up to 77 eggs. Now that's still not too crazy compared to the 3 million eggs that we could see in an average brood. But this is an extreme case, so we're going to take the small end of that and assume there's 750,000 eggs in this brood. So we're going to also take the max intensity, which was 204 worms per brood, and assume all of them are eating at this really high feeding rate of 77 eggs over the 17 days. In total, that'll be about over 170,000 eggs eaten over that 17-day development period by these worms. That's up to 22.8% of the blue crab's brood, which in the grand scheme of things might not be a lot, but it's a notable portion of the reproductive output of the female. And although this is an extreme case and it might be unlikely, it doesn't mean it can't occur. It's not impossible to occur naturally. However, we did see other ways that these worms might be harming egg mortality in blue crabs. During our experimentation process, it was not uncommon to see a worm knock an individual embryo off a CD strand, which is what that line going through all those embryos, it's holding them together. And once an egg is knocked off that strand, it is very unlikely for it to develop properly into a blue crab zoea, which is its next stage of life. We also very quickly learned that they're like to leave a lot of debris behind and ditch their little mucus homes that they build for themselves, which is what you see on the left here. And all of this put together makes a really great environment for microbial growth, which will eventually migrate to the eggs, which as you see here, we get a lot of fungal growth, which impairs their ability to develop properly. So overall, what I want you to take away from what we've kind of found here is that although nemerteans are not a major source of mortality for blue crabs, at high densities, they could be having a notable impact on them. All in all, that the blue crab is facing a lot more impending issues such as overfishing and the loss of nursery habitat. So the worm might not be putting up a fight for being the culprit of the decline of the Chesapeake Bay blue crab, but it's having an impact on egg mortality on a whole. But as you saw, there's a lot of variation in those data cases that I showed you, anywhere from 0.001% of the brood being impacted to 22.8. So there's still a lot we don't know, but we're starting to put the puzzle together. So this was a good step. There's lots of other things for us to figure out though. Some things that I'm interested in potentially inquiring about next is salinity tolerances of the worms, seeing if there's potentially a refuge point for infected females in the bay, and also looking into what accounts for the variations in feeding, since we saw anything from eating 0 worms over that 6-day period to anywhere to 27. There could be anything in close to being their reproductive habit. There's not a lot known about the life cycle of this species of nemertean worms. So we have lots of places to go to kind of piece this puzzle together some more. But until we have answers to those questions, I would like to take some time just to thank everyone who made this summer such a wonderful experience, and for everyone who was a part of building this program to make and support undergraduate research, NSF for funding us, and a special thanks to my advisor, Alex Schneider, for being a well of wisdom, a backbone of support, and making my overall experience here at VIMS what it has been for the past year. So thank you all for your time, and if anyone has any questions, feel free to ask.
Wow, thank you for that great talk, Alex. We'd love to open the floor to any questions at this time.
Well, there is a question that came in from the Zoom chat. Is there anything you would do differently next time in your experiments?
There's a couple of things. So we actually ran a pilot at the beginning of the summer that caused us to alter a lot of our experimental procedures because we found microbial growth to be a real strong factor that we had to combat. And so we did change a lot of our experimental procedures in terms of sanitation. I think moving forward, we would try to run our eggs through a really low bleach solution before putting them into the well to make sure they're not bringing in any fungus or microbes with them when we take them from the brood. So I think that would definitely be a first step.
Thanks. Has anyone else done feeding trials between with nemertean?
Yeah, so Pam Rowe did them on a different species of Carcinomyrdes in, I think it was about 10 to
Well,
20
where I
years.
was heading with this
Okay.
is how do you know that the feeding rates that you observed are actually natural feeding rates?
Right. And so I think that's an issue that comes with any lab experiment, taking an organism out of its natural environment and putting them through the stress of bringing them out of a brood that, so we're not stimulating perfectly natural environments, but this is a similar procedure. And so this is the best information that we have at the moment. And so hopefully we're able to build a little bit better information. I definitely think it's something that should be rerun. Go for it.
So how do these compare with those that Roe did?
Right. So that species feeds very differently, not in terms of anatomy, but this species, we don't have much to compare it to because the species that we see in the crab that she studied caused the collapse of a fishery, I believe, or so it was having a larger impact on the crab that it was seen in than the ones that we see here. And so their feeding rates are going to differ in that capacity just because we're not seeing it on that max value too on the broader scale. I think that's the best way I can answer that right now.
Yeah, another question here.
So thanks, Alex. That was great. I'm wondering about the crabs that had the worms and those that didn't, and whether you could see any differences in the health of those crabs, because you mentioned that the worms may have other negative impacts like on bringing in bacteria or other things. So I'm just wondering overall health, could you tell anything?
Right. So we do have data about the condition of the crab that was on the worms. We haven't done analysis on it yet, but just from looking at them, I honestly, those that are newer and like cleaner that might have just gone through their terminal molt tend to have less worms, just from looking at it and being a part of the dissection process. And so as you can see, some of them tend to accumulate worms in their gills as well, and those can migrate back out into their brood. So that might also be a factor of how many brood and spawning cycles the female has gone through, could be a factor in how many worms that she has as well.
So you'll be looking at that later?
Yeah, so there is actually a paper that was published about using nemertean worms as potential biomarkers because they migrate between worms, their gills and their broods. So that is definitely something we'll be looking into.
Great, thanks.
Yeah.
Another question over here, Stephanie.
Absolutely fantastic talk. I had a question. I know that the females get the nemerteans from the males just after their terminal molt, right? How do the worms get in the males?
So we're not entirely sure that they're being transferred from male to female.
Oh.
So we're not sure if the larvae are actually potentially recruiting straight into the brood or the gills of the female. So that is a potential. We don't know enough about the life cycle of this species to say they're migrating from the males to the females yet. Yeah.
Any other questions?
I actually have a question. So in some of the oyster experiments that my lab does in getting respiration rates for oysters, we put the oysters, before we measure respiration, we put them in a brine so that the worms for oysters, they don't like that brine solution. And so they escape. So it's easier to get rid of them so that we can get a true measurement of oyster respiration, not a combo respiration.
Okay. So
do you know anything with the nemerteans?
Yeah.
Their salt range tolerance?
Right. And so looking at salinity tolerance, so I have actually, that's kind of the next step. I'm going to be doing a thesis throughout the next year. And so we have plans to look at salinity, and I've done some preliminary trials on it. And I have I've shocked them into 10 PSU, and I've also done a really preliminary mortality study at 10 parts per 1,000. One of them did die, but the rest of them seemed to just not be in great condition, but they were able to survive through that. And that is kind of the low end of where a female would migrate through. And so looking at that, I'm not sure if that means that they're leaving the crabs or if they're just unhappy when a female's at 10 parts per 1,000. But yeah, they seem to be pretty resilient so far. So yeah, of course.
All right. Well, with that, let's have another round of applause for Alex. Thank you, Alex. Ladies and gentlemen, thank you again for attending these talks today. We'd love to have you here, and thanks for coming to support our REU students. We've made it to our final talk for today, and our next presenter is coming all the way from California State Polytechnic University, where she is an environmental biology major. She's going to talk to us today about stingray foraging holes. And she's the only REU member from the West Coast. Ladies and gentlemen, last but not least, put your hands together for the West Coast Queen, Tiffany Huang.
Thank you, Eli, for that introduction. Like Eli said, my project is on the effects of stingray foraging holes. And this summer, I had the honor of working with Dr. Annie Hensel and Dr. Christopher Patrick with the SAV lab. So a little background on my project: coastal ecosystems like seagrasses are formed by living organisms and are structured and maintained not just by their physical surroundings, but also by their local fauna. Seagrasses are important to us because they provide important ecosystem services. They are a nursery and foraging habitat for many important species like the bay scallop. They also trap sediment, which improves water quality, and they provide ecological services like carbon sequestration, which is the process of removing and storing CO2 that's in the atmosphere. Unfortunately, seagrasses have been declining worldwide in the last several decades, and because of their importance, restoration efforts are becoming a top priority. To properly conserve these meadows, we need to understand the natural processes that form and maintain these ecosystems. One example of a natural process ties back to the fact that because seagrasses are a living habitat, they are equally influenced by the fauna and physical process. Seagrass heterogeneity or patchiness is not only shaped by physical processes like hydrology, but also by the fauna that resides in them. For this project, I focused on stingrays as they are large-bodied fauna that can change seagrass heterogeneity. As stingrays forage for their prey, they change seagrass heterogeneity by either suctioning onto the sand and huffing and puffing, or by coming in and digging and uprooting seagrass and sediment. These activities create bare depressions in the sand with torn and exposed seagrass edges, and these holes can range anywhere between 1 to 2 meters wide. Therefore, these holes can create heterogeneity or complexity in what would have been a continuous seagrass bed. Although ray foraging is a natural behavior, the relative role these foraging holes have on seagrass top-down and bottom-up processes is currently understudied. There have been a few studies that have shown that small-scale seagrass disturbances like ray holes can alter top-down processes like predator-prey interactions. However, among these studies, the results have varied. Also, since foraging directly removes seagrass, this may change certain ecological bottom-up services like carbon sequestration. And like I said before, since seagrasses are declining worldwide and restoration efforts are a top priority, it is important to have a better understanding of the natural processes that alter seagrass functions and services. So here we measure how ray holes affect predator-prey interaction and bulk carbon storage. To quantify the difference in predator intensity, we used a tethering experiment with live prey, and to quantify the difference in bulk carbon, we collected benthic cores and muffled them in the lab. We hypothesized that predator intensity will be greater at holes compared to seagrass beds because prey will be more exposed. And we hypothesized that bulk carbon storage, our proxy for carbon sequestration, will be lower in ray foraging holes compared to undisturbed continuous seagrass because of the direct removal of seagrass and the increased susceptibility to erosion. For our experiment, our study site was a dense seagrass bed with over 70% grass cover in South Bay, which is located within the coastal bays of Virginia.
Prior to our experiment, we conducted snorkel and boat transects to locate bare patches that showed signs of ray foraging behavior within seagrass. And for the purposes of my project, we defined ray holes as a bell depression at least 1/2 to 2 meters wide in the sand with torn seagrass edges. The waypoints up here show you where we collected our data. We located 10 holes in the north side of the bay and 10 on the south side of the bay within a known dense seagrass bed. And for our seagrass plots, we simply located a full bed of seagrass at least 2 meters away from the paired holes, and all of our paired plots were at least 5 meters away from each other. Once we located all of our ray holes, we used a paired design to measure the differences in predator-prey interactions and bulk carbon in ray holes and their adjacent seagrass bed. We tethered prey to understand how predator-prey interactions were altered by the presence of a ray hole. We had 3 different types of prey tethered onto monofilament long enough so they could move around in their natural behavior. For our prey, for those of you who are curious, we tried to mimic what would be naturally available, and we placed priority on those that would be of interest to rays and also ones that were readily available to us. Initially, we were thinking of ways to represent bivalves and mobile prey, and we ended up using clams, scallops, and worms. A local clammer offered us 3 to 4 inch greater clams, and the VIMS Scallop Hatchery up at the Eastern Shore Lab donated 3 to 4 inch base scallops. And unfortunately, our initial hopes of having local polychaetes and juvenile crabs were not possible, but we used earthworms as a proxy for soft-bodied mobile prey. The prey items were either tied or superglued onto monofilament and secured into the ground using garden stakes. After a 24-hour soak, we recorded the prey type missing and assumed it was eaten. And we repeated this process over the course of 3 days to try to get a broader picture of the predator-prey interactions happening in the bay. To identify predators and potentially get predation rate, we randomly selected 5 pairs to record for 2 hours, and we repeated this over the course of 2 days. And for our bulk carbon, which was our proxy for carbon sequestration, we cored 10 paired plot plus 10 paired hole plus seagrass beds independent from our tethering experiment. A core was inserted vertically down from the seagrass bed's natural surface, and for our ray holes, we kept the top of the core level with the nearby seagrass to collect the amount needed to compare what has been lost. To estimate the amount of carbon in our samples, we used subsamples of organic and inorganic material, and based on known field methods, we conservatively estimated that around 40% of the organic carbon measured, that of the organic material measured was carbon. And for our plant biomass, we estimated conservatively that about 50% of it was carbon. Lastly, for our analysis, we used linear mixed effect models where our pairing and our day was our random effect, and our model assumptions were diagnostically tested using the R package performance. I'm first going to go over the results of how ray holes potentially altered predator intensity, and I have 2 main results to review. First, we simply compared plot types, which throughout my entire presentation will be holes and seagrass, by the proportion of prey type eaten after 24 hours, and we categorized the results by clams, worms, and scallops. And because we repeated this experiment over the course of 3 days, we will see individual points representing the average for that day. When we looked at what was consumed, our experiment showed that ray holes had a significant impact on what prey item was consumed. Specifically, clams were basically never eaten in holes or seagrass, while worms were always eaten. But ray holes did alter scallop predation, where scallops were eaten around 25% more in seagrass than in holes, and I will get back to this at the end of my predation results. To further dive into potential changes in predator-prey interactions, we used GoPro footage to look at what predators were present and to see if their activities were altered. Briefly, we had about 8 individuals, and here are some examples I was able to get. We categorized them into fish like this spot and pinfish, and the others were categorized into scavengers like this hermit crab and black-fingered mud crab. While I don't have the formal results to share here, we did observe that larger body crabs like the mud crab had altered activity levels by these ray holes. We did test if predator encounter rates were altered by ray holes. This graph compares plot type by predator encounter rate in minutes, and we also further categorized the results into clams, worms, and scallops. Ray holes appear to alter how often worms or soft-bodied and high-motion prey items were encountered. Worms were encountered 25% more than clams or scallops, regardless of the plot type, and worms were encountered roughly 3 times more in holes, although due to high variation, this was not statistically significant. Our mobile prey item really allowed us to see how ray holes altered how often prey were encountered by these predators. Upon reflection of these results combined, ray holes may reduce predation from larger bodied crabs. Specifically, our results indicate that the refuge of the seagrass allowed for larger crabs to take their time to prey on our items. Larger crab species like this mud crab were not seen very often in the holes, but we do have footage of higher activities in the seagrass. This gives some insight as to why 7 scallops were consumed in the seagrass versus only 1 being consumed in the hole. Because crabs need more handling time to crunch the shells, and because of that, they may need the protection that seagrass provides. In summary, ray holes only altered scallops being eaten, and ray holes also altered how often mobile prey were encountered. Now, we looked at predator intensity to look at top-down processes altered by ray holes. And for our bottom-up processes, we took sediment cores to look at bulk carbon. The graph on the left shows carbon estimates in grams in just sediment between holes and seagrass, where seagrass had about 1.2 times more carbon than holes, although this was statistically insignificant. The graph on the right, however, shows carbon estimate also in grams, with both sediment and plant biomass between holes and seagrass, where seagrass had about 8 times the amount of carbon. Just to conclude, and as a reminder, we compared how ray foraging holes, a natural disturbance, altered top-down and bottom-up processes in seagrass. Specifically, we found that their foraging behavior did alter predator-prey interactions within the bed, and locally, their holes greatly reduced the amount of estimated bulk carbon. In considering seagrass restoration and conservation in the bay, our studies indicate that small scallops might be more vulnerable in continuous seagrass. And this finding is important because scallops are an important fishery species, and there are hopes of having them reintroduced in the Chesapeake Bay in the near future. And for the sediment core surveys we conducted, we will couple them with a South Bay-wide ray hole survey to scale up and estimate the amount of carbon lost due to ray foraging. Specifically, our bay-wide hole surveys will be done on the ground. However, I do have an aerial image that potentially shows you how many ray holes there are in the bed as of May. To give you a little insight, here's the whole bed again. And as you can see, here are the 2 sites where we collected our data. I just wanted to point out a couple of examples of potential ray holes. And I wanted to show that there aren't just a couple of ray holes in the bay, there's potentially enough to completely change the complexity of a bed. To conclude, in the broader context of seagrass conservation and restoration, our study shows that natural disturbances like ray holes
play a role in altering species interaction and ecological services. These findings can be considered in the first steps of restoration when restoration practitioners consider what size the bed should be so they can take advantage of ray holes, creating spatial complexity, as opposed to completely destroying all of their efforts. I'd like to thank some people involved with this project. Thank you to Hayden Akers for her amazing assistance on the field. We could not have done this without her. Thank you to Johnson and Kirkland Lab for letting us use their material for our bulk carbon. Thank you to Reba T. Smith, Bowden Lusk, and the Cherrystone Creek Clam Company for donating the prey items used in our tethering experiment. And we would also like to acknowledge our funding source, the VIMS REU program, and the National Science Foundation for making all of this possible. Thank you everyone for watching.
Thank you for that great talk, Tiffany. We'd love to open the floor to questions.
Thanks, Tiffany. I had a question about your prey that you used for your tethering, specifically the earthworm. Were the earthworms aquatic? Could they breathe underwater?
No, yeah, no, we just use like regular earthworm bait from the store.
They've just died underwater and it would have been scavenging?
We kept them alive, like right up to the point where we put them underwater. Like we just tried our best. We couldn't get local polychaetes because they weren't big enough to tie with the monofilament. So we just wanted something that was like soft bodied and like they were like based on the GoPro footage, they were kind of alive for a little bit and their like movement attracted the fish. So yeah.
Okay, thank you.
Any other questions? Yeah, we have one here.
So from the tethering results, particularly on the scallops, showing them more susceptible to predation within the seagrass as compared to the holes, how can you, or how would you suggest scaling that up to figuring out how to restore these beds? Because that almost would suggest that you don't want as much seagrass.
Okay, great. Yeah, great question.
How do you rectify that?
Yeah, so the results on like scallop predation kind of vary based on like the literature that I've read. Our scallops, the scallops that we use were kind of on like the bigger end of the scallops in like the studies overall.
How big were they?
The ones in the literature that I've seen, they were more like 1 to 2 inches, while ours were 3 to 4
inches or centimeters?
Inches.
Are big scallops.
Yeah.
Okay, thanks.
Any other questions?
Who had a question? Okay, I do have a question. Can you go to your slide with where you do the sediment plus the seagrass carbon? Yeah, so that one. And I think it really hit home when I saw that satellite image of the number of holes that a stingray can make. So from the world of, now taking it from a scallops industry to a global carbon industry, which is my area of study. So a lot of the coastal ways, a lot of our global carbon budget models show that the coastal waterways are a huge sink of atmospheric carbon. And I think this plot on the right really shows, the contribution of seagrass to the carbon budget. So if you have a bunch of stingrays going into an area, how drastic of an impact, like how many holes can they really or how much seagrass can they really wipe out? And do you think we might need to readjust our carbon budgets to take that into account?
Yeah, so our ray hole surveys aren't completely done yet. I don't have an image up here, but we do have a site that we call like the Ray Party because it was just like completely destroyed. The seagrass was completely destroyed. I don't know, like if they just had a fun time feeding, but yeah, I definitely think once we get the information from the ray hole surveys, I definitely think that I mean, since like we already see like such a drastic difference in just like a local scale, I definitely think that we should have another look at the carbon budget.
Awesome, thank you.
Awesome talk.
Thank you.
I've got a question that kind of follows up on something that Rochelle was asking about. So how long are the tethers for the scallops?
They are 20 centimeters, at least 20 centimeters.
Okay. And scallops are pretty mobile, at least they are capable of jetting around when they're not tied down. So in a natural setting, do you think that the predation rates on the scallops, if they weren't tethered in the seagrass, would have been as high as what we observed?
That's a great question. I want to say I don't think it would be as high, just because when I was reviewing the GoPro footage, I saw a lot of the scallops moving around, and because of the tether, they were kind of like stuck in the same place. So I assume that if the tethers weren't there, then they would have been able to avoid predation.
So more of an encounter rate with these predators rather than an actual mortality rate, perhaps.
Yeah.
Cool, thanks.
Do we have any other questions for Tiffany? That's great. Well, thank you so much, everyone, again for your time. Let's hear another round of applause for Tiffany and the RE presentation.
All right, that brings us to the end of the program, but I want to make sure mentors stick around for just a few more minutes. We're going to have a little reception right outside in the lobby in just a few minutes. And I wanted to take time right now just to once again thank all of the mentors for all of your input on the program this summer. I know the students really appreciated it, and I'm just amazed at these presentations we saw today. Great job, everyone, and great job to the mentors. Round of applause for mentors. While we're giving out thanks, I also want to say thank you to our two session chairs. So Eli and Sarah, thank you so much. Once again, I wanted to thank the other assistants in the program from Stephanie Peard and Grace Massey, because you really pulled it all together, and we had the great team this year, got it done for the first time in person in 2 years. So great job. And so with that, just a couple of things for the interns. Don't forget, you still have those papers to complete. So we'll be looking forward to seeing all of this that you presented in print. And also there are a couple other things. There's the post-program survey that you need to fill out so that we can improve the program and take your ideas to make it better for the next cohort. And then also the checkout sheet, which is a VIMS thing. Everyone has to do that. So please don't forget about those things. And then Grace has a couple of, well, the completion certificates that we'd like to give each of you. So if you'll stick around for a minute. So in no particular order. Okay, Sarah Vadachar, come on up. Oh, and you get, everyone gets a certificate of completion that you can hang on your wall, plus a bookmark. And there are some books outside in the lobby that have an illustrated guide to deep sea creatures. So that's for everyone. All right, Eli Whitehead Zimmers. And Garrison Bullard. Okay. Okay. Okay, and our next one is Zoe Galvin. Next we have Tiffany Huang. Grace Levins. Nice job. And James Liu. We also have Cole Miller. King of crabs. And Alexandra, wow, Alex Pomeroy. Anissa Sellers. Great job. And Lindsay Smitty Smith. And we also have Grace Kirkpatrick, who is out in the field today. So with that, I want to say thanks again to all of the interns, and we'll look forward to seeing those papers. And please come join us outside for an informal celebration. Nice job, everyone.